Well, I think we may as well make a start, at least in the first few minutes. My name's Nick. Today we're hosting a hands-on modelling session workshop hosted by the British Toxicology Society. It's a free event via Zoom, and we'll be recording the session as well. This is the first event of its type where we're bringing together two speciality sections of the British Toxicology Society. We're bringing together our friends from in silico safety science and also mechanistic discovery toxicology science. The ultimate scope of the workshop is to hopefully foster a community where we can bring people together, not necessarily from a mechanistic and or an in silico toxicology background, but also we bring together people to help tackle questions, particularly around chemical safety and toxicology. So whether that might be applied to the drug discovery setting, might be applied to the agrochemical setting, any setting ultimately that hopes to use chemical structure data and data that's already been deposited elsewhere to help answer questions ultimately. With the focus of hopefully arming you guys with the ability to explore chemical structured data, promote an understanding of programming and artificial intelligence and machine learning tools, particularly for those that are not familiar with the use of these tools. And also to guide people towards the idea of using in silico tools to perform hazard identification and risk assessment in the toxicological setting. And the ultimate aim of this workshop is to kind of almost remove any barriers or what you might call disconnects between those people who are purely in silico scientists and those who are what I might class as like a thoroughbred mechanistic toxicology or safety expert. It's maybe clear at the moment that people using in silico tools are not necessarily the people making are making the decisions in a, say, a drug discovery scenario on the basis of a risk assessment. So it's to break down those barriers and hopefully empower people to be able to thoroughly understand not only what these models can do, but ultimately where the data is coming from, how that data is assembled, and ultimately how we can use these models and modalities to help us answer complex questions. And of course, this has a particularly big driver is of course is the 3 R's. So that's the reduction, the refinement, and ultimately the minimizing the use of animal studies in the toxicological setting, and of course the idea of kind of novel approach methodologies to support toxicology in the future. So whilst we're not suggesting that computational or in silico tools are necessarily going to replace animal models anytime soon, despite the act that was passed by the FDA a few months ago, we're not stopping the use of animal models in the drug discovery or toxicology setting. Moreover, we're now allowing other tools to be used that can support those in vivo data packages. And of course, that's where these novel approach methodologies really come into their own. So today we have, ultimately we have 2 groups on the BTS, and ultimately we have 3 people with us from computational and in silico toxicology specialty groups. We have Professor Andreas from University of Cambridge, Srijit, who is a doctoral scholar at University of Cambridge, and we have Layla as well, who has recently just completed on her PhD at University of Cambridge, and now is a co-founder of a new company called AbsoluteAI. The general overview of today's workshop is Andreas is going to give us an introductory lecture around the fundamentals of predictive modeling of small molecule properties. So hopefully this will give you guys an insight into how we define small molecule chemical fingerprints, what it means to model, some of the terminology that you might come across in this session, and also that you might have seen in publications, for example. So what is a random forest model? How are some of the models derived that are currently being published in the world of in silico toxicology? And then ultimately, we're going to move into 2 hands-on exercises that are run in something called Google Colab. So this is an environment that everyone on the call will run in their own web browsers. We'll show you how to do that shortly. But ultimately, we're going to take you through some programs that Layla and Srijit have written, and they will take us through some hERG bioactivity data. So in a drug discovery situation, inhibition of a hERG by small molecule leads to ventricular repolarization, elongation of the QT interval, and ultimately that leads to cardiac arrhythmias in the clinic. And the second exercise is around a classification model based on mitochondrial toxicity data. So again, this is data that's been curated from this publication here. These are all based on simple endpoints. So it could be IC50 for IC50 values, for example, and we'll be using basic physicochemical properties to help define some of those data sets. This link here you'll all want to take a copy of this afternoon. This is the link that will take you straight to the Google Colab environment for the sessions this afternoon. All of these sessions are being recorded, and eventually they'll be deposited via a YouTube channel, which you guys can go to using this handle here, Andy Dendy. We'll post we'll send this link round as well at the end of the session, and it will get sent around by email as well. Throughout the session, obviously, there's quite a few of you on the call, and we appreciate there's probably going to be an awful lot of questions at any one point. So if you could, when asking a question in the chat window, if you could use either #technical or #general, we'll do our best to kind of respond to any of your questions as best we can. And again, at the end of the sessions, there might well be opportunities to ask questions orally. So I want to make this clear before I pass on to Andreas that any statements that any of the organisers make during this workshop are in our capacities as academics and or members of the British Toxicology Society, and not necessarily of who employs us or where we're going on to work. So with that in mind, I'll pass on to Andreas. And I think you've now got control, Andreas.
Okay, great. Can you see that and hear me, Nick? Huh?
Yeah, I can see that. All good.
Okay, very good. I'm just uploaded my slides to the website as well. So in case you would like to read through them in detail or check references and so on, feel free to download them from that link here. I just pasted in the chat as well. Yeah, so what we will start with today is, and this was advertised explicitly as a workshop for non-experts, right? Non-experts in modeling, but many of you will have a toxicology background and so on. So, so I will use terminology that's as accessible to experimentalists as possible, but very happy to clarify any open questions. I was actually working on that interface between computer science and life sciences for, I don't know, 15 years. And in Leiden, 15 years ago, we wanted to establish an interface between the pharma IT platform, it was called, between computer scientists as well as life sciences experts. Yeah, so I'm very well aware of that. That is actually, it's not an easy field, right? You start thinking differently if you study, right? That forms how you think. But actually, I realized it's very important to understand chemistry, biology, as well as machine learning. Yeah, otherwise you cannot answer relevant questions. Yeah, and that's actually, as Nic illustrated already, what we are trying to do here, to bring experts from different disciplines together, because together it's then more than the sum of the parts, right? Very important to understand other domains in order to do something meaningful with a computer. Okay, and so what I will go through today is now, I will present fundamentals of how do you build machine learning models for life science endpoints. The whole thing is simpler than you might think, and so I will go through that stepwise, and it should be easy to understand for also general scientists and so on. As Nic said, so I'm talking in my capacity as an academic. We started companies as well, Pangea Botanica most recently, but here it's about the science and how we can model data. So I'm talking as an academic here. So there will be 3 parts in this presentation. The first part will be on data and endpoints. What matters is what we put into the model, and that determines really the applicability domain of the model. Which types of chemistry can we model? What is the endpoint we choose to model, and so on. And the data we put into a model is key. We can think about it in terms of a hierarchy. First comes the data, then comes the descriptor. This is how I describe the data I have to the computer. I'll go into that in a few slides. And only finally comes the machine learning method. That is usually the order of importance as well, okay? How much data do I have? What's the quality of the data? That comes first, and that's what I need to ensure in the first instance. And then there's the question, how do I actually formulate? How do I describe the problem to the computer? And then there's the machine learning method. And quite often right now, there's of course lots of focus on machine learning methods, deep learning, and so on. I will explain what deep learning models, neural networks are as well. You will see it's simpler than you think. There's quite a lot of focus on methods, right? But that's usually only at the end of treating data, right? Dealing with data. It's actually in that order. Which data do I put in? How do I describe it? And then comes the machine learning method, okay? And that's the order of importance. I've seen in virtually all projects I worked on how we need to address that problem. So we'll talk about data, then models and model validation. That's a tricky topic as well, in particular in chemical space. I will illustrate that because chemical space is large and very diverse. And then there's also a few, lists to take home, basically. If in the future you talk to a computer scientist and tells you that the model predicted X, then there's a few questions that actually come from the preceding sections of this presentation. There are a few questions that you can ask your friend, the modeler, right? In order to estimate how good the predictions of that model actually are. Yeah, it's not a black box that spits out a number, right? There's a process how a model gets established based on the data you put in, how you validate the model, and so on and so on. And based on that process, you can actually estimate how good the predictions likely are. Okay, yeah, so that's what we'll go through the next 40 minutes or so. First of all, there are 2 types of models, okay? 2 very different types, and the terminology overlaps sometimes a bit, so it's important to take that apart. You can actually model in 2 different ways. First, you can have an understanding of the system that you want to model. Let's say gravity, for example, right? We have a mathematical functional form, how to describe that. Or if I think about proteins and bonds, for example, atoms and bonds I want to model, I can use so-called force fields, that use Newton mechanics, basically, in order to describe the dynamics of the system. And in those cases, I have an understanding of the underlying model. I have a functional form, often that's mechanistic. If you come from the toxicology area, for example, adverse outcome pathways go a bit into that direction, but they're not fully mechanistic, of course, but they are pseudo-mechanistic. That's often how I call that, right? On other cases, you have actually, you have a particular a biological signaling chain, for example, where a particular modulation of one kinase, let's say, leads to up or down regulation of a particular gene in the end. So in those cases, you have mechanistic models, okay? And in those cases, you actually have an understanding of the system, you have a mechanistic functional form, and that means 2 things. Firstly, it means you need to understand the system in order to establish that functional form. And that is the problem you're confronted with. But once you have that, you're able to model novel systems, okay? You actually, let's assume you have a protein dynamics, you understand how protein dynamics works, then I can model any protein, right? Not only one protein, I can model any protein, okay? So once I have understanding of a system, I can do a model-driven treatment of a system. That's one way. In many cases, though, we don't have that understanding of the system. And in those cases, I need to model data empirically. So we do not have an understanding of which mechanisms of action, let's say, lead to a particular phenotype. And in many cases, that is true, more often than not. And in those cases, we need to basically create data, generate data. So we need to measure, let's say, the bioactivity of compounds in a particular assay. And then we empirically establish which compounds lead to a certain safety or efficacy relevant endpoint and which compounds do not, okay? And in those cases, we can build a computational model based on, say, chemical structure as input, and we have the phenotypic effect, efficacy or safety related, as output, yeah? And that is purely empirical modeling. So we do not require any understanding of the system. This is a data-driven model. And that means two things. First of all, we don't need to bother with understanding the system in the first place, because all we do is empirical modeling of the system. So that makes it easier in the beginning. However, the trade-off is that the model is only able to capture data that we actually generate ourselves. So we can model that data, and then the model ideally can extrapolate to an extent, but not perfectly. Yeah. So if we generate empirical data related to chemical space and a safety endpoint, our model may be able to extrapolate to an extent to novel chemical space, slight modifications of the molecule, right? But it will not be able to make predictions for completely different chemical space. And that is because we don't understand the underlying mechanisms. We don't understand the biology in many cases of the system, okay? And these are two fundamentally different ways of modeling. Understanding the system, you can model any system, but requires more work upfront. Or data-driven modeling, in those cases, we empirically model our system. It's easier to do. We just have a machine learning model in many cases, but it doesn't extrapolate so well, okay? And many of the models that are around right now, when it comes to biological endpoints, are actually more in the data-driven domain. You also have some areas in the middle, and that means combining prior information, network information, let's say, as well as data, right? But these are the endpoints, basically, or the extremes of generating a model. You can either be model-driven or data-driven. And much what we discuss here will be in the data-driven domain, okay? How can we empirically model relationships between a chemical structure and a particular safety or efficacy endpoint? So what is a computational model? So what is shown here is basically, if we do an experiment, right, we put in a structure, tamoxifen here, for example, and we measure an activity in a particular assay. So there's quite a lot of conditions that are associated with the generation of the data. So we measure the activity of that compound in a particular assay setup, and we obtain an IC50 or LC50 or whatever bioactivity endpoint we want to obtain. So we have an empirical relationship between a chemical structure at a given dose, given a particular assay setup, and a particular bioactivity endpoint. Okay, that is the experiment. So what do we do if we model computationally? There's mainly two things that we need to put in between the molecule and the endpoint. That's what we do computationally. We need to, A, describe our molecule using so-called descriptors. Think about it, for example, in terms of molecular weight, substructures, and so on. I will come back to that in a second. But you need to describe your molecule to the computer. And the second step is to build a machine learning model that uses a combination of descriptors to predict the endpoint. Okay, so a very simple example is descriptor, for example, you have a log P, let's say, in molecular weight, yeah, and you build a model for blood-brain barrier permeation, and you know that certain lipophilicity ranges are actually favorable to get into the brain, right? You need to have a certain lipophilicity, and molecules need to be small, molecular weight below 300 or so, right? That's a very simple case where we have molecules, we describe them using molecular weight and log P, let's say, or log D, and then we have a model that in this case only combines two factors in order to predict whether a molecule gets into the brain. That's a very simple example. Of course, we can have higher dimensional descriptor spaces. There are many ways to generate descriptors, but the concept is the same. Molecule, you put it into a numerical or presence-absence binary descriptor form for the computer, and then you fit a model in order to predict the endpoint. That's a general concept. So what are limitations of models? And that's very important, especially in this data-driven domain that we're dealing with, right? Models only know and what gets explicitly described to the model. Okay, sometimes there's a perception, okay, AI solves everything. Well, it won't. It won't, because it's based on the data. Any computational system is based on the data you feed in. Okay, What are the limitations here? In input space, if you have a model for any safety or efficacy-related endpoint, you're limited by the chemical space that you actually have data for. Okay, completely novel chemistry will be very difficult to predict. So the chemical space coverage that you put into a data set, that is all that is available to the model. And models don't really reason beyond that data. To an extent they can, because the descriptors you choose hopefully allow the model to detect underlying relationships. Yeah, if I have 20 different data points of molecules that get into the brain and don't get into the brain, hopefully the model picks up underlying trends in the data and finds out that, okay, smaller molecules in a certain lipophilicity range, they get into the brain, right? And then in that descriptor space, you can extrapolate to an extent. But by and large, that depends on the number of data points you have and whether you choose suitable descriptors in the first place. Okay, so the model is limited to the chemical structural space I put into the model and the ability to extrapolate within reason within the descriptor space that you actually use in your model. Yeah, so that is the second point here. So if chemical space you put in, the model is limited to that, can extrapolate in the descriptor space, assuming that is a reasonable descriptor space. For the example, blood-brain barrier permeation, for example, substructures would probably be a less good way to describe the system, right? If molecular weight and log P or log D are actually pretty strong determinants whether compounds go into the brain. Finally, also, and that is a major problem actually when it comes to machine learning right now, you also need to label data. Yeah? There are supervised and unsupervised models. Supervised models, you fit basically your model on a particular numerical or categorical or binary endpoint. These are supervised models, where you have input data and you fit it to the output data. And you have unsupervised models. And unsupervised models are clustering, for example. Okay, and you need to label your data. And that is very tricky. I'll outline that on the next few slides with many in vivo relevant endpoints. Okay, and these are actually tricky parts of building a model. Do I have chemical space coverage of the compounds I'm interested in? Do I choose the right descriptors? And can I actually label my data? Okay, I'll come back to all of those points on the next few slides. But basically that means that many of the approaches that work very well for image recognition and so on, they don't automatically translate to the chemical area. Because this chemical space question, do we sample that well enough? It's large, do we have enough data? It's quite specific to our area. Describing molecules is quite specific, and also whether the endpoints translate to the in vivo situation, okay? And that is also not always true. So let's come to the topic here, data and endpoints, the coverage, conditionality, error on the data, predictivity. So this is a slide from before, right? So we have experimentally a compound linked to an endpoint. That's what we measure, and the whole measurement is conditional, dependent on the assay setup, concentration, and so on. And what we do in the computer is we convert the molecule into a descriptor, we fit a model, and we predict the endpoint. So, and there are two different situations, two different types of data that we can have when we model life science related data. And these are what I would call early stage proxy space, left-hand side here, and the more in vivo related space. And that's a very difficult topic that, in my opinion, gets neglected quite often when it comes to AI in the life sciences area. In proxy space, so which type of data is that? Log P, log D, biochemical assays, for example. So these are data points that followed logically from, let's say, the era of high throughput screening, right? So when it became fashionable to generate large amounts of data, million data points or so in a high throughput screen, that then needed follow-up assays as well, right? So you then have, that means we're now in the situation where we have large numbers of data points, tens of thousands or hundreds of thousands of bioactivity data points. But in many cases, these data points were generated for in vitro systems, right? Quite far detached from the in vivo situation. What is characteristic for that type of data? Well, we have lots of data because it's cheap to generate. And we can also label that data relatively well. Why is that? Because the data is largely unconditional. If I have let's say a log D model, I have a isolated protein in a biochemical assay, yeah, and I measure a KD in that system, dissociation constant, then that is by and large, actually, that's a number that's thermodynamically defined, right? And that's a number that's largely independent of any of the assay setup, right? Of course, there are trickier areas as well. I have a kinase assay, my IC50 depends on the ATP concentration and so on. So it's a grayscale in between. But in vitro data, if I think about that in the extreme, we have a large number of data points. We can label them so we can build machine learning models. Okay, but it's far away from the in vivo situation. On the other hand, and that's usually what we deal with in safety space on the right-hand side here, I have quantitative data. So effects depend on dose, exposure. I have complex models, often in vivo models. I have histopathology endpoints, where actually we need an expert because we have small numbers of animals, where we need an expert to say whether a particular endpoint is treatment related or not. So we have much less data. It's very conditional data. Things depend on dose. And that is actually much less favorable from the machine learning angle, right? We have fewer data points. We will see later that is important. We have fewer data points and we have very conditional data, okay? But the data is more in vivo relevant, okay? And that's one of the big problems right now when it comes to using data in AI systems in order to make in vivo relevant predictions. In supervised models, we have input data, descriptors of the molecule, and we have a supervised method that predicts the output, let's say an IC50, okay, against a particular protein. But the areas where we have lots of data, it's usually in vitro data, so quite far away from the clinic, basically. That's what we can model. When we go to the in vivo area, we have much less data, right? It's very conditional data. And that's one of the problems we are right now confronted with, okay? And the questions that are asked from the data, they actually, well, we ask in the areas where we have more data. In the in vitro space, we're asking quite often the simpler questions. It's a discovery setting, virtual screening, find me some active compounds, right? And any compound fulfilling a set of criteria will do. Find me any active compound out of 10 to the 8 compounds I could buy, for example. Give me any of the 10 to the 5 compounds or so that are bioactive. It's a simpler question. There's many options to succeed. Efficacy or safety is much more difficult. We need to make a prediction for a particular molecule quantitatively based on less data, right? As I've shown before, very conditional data. And there's a long list of criteria to roll out, right? Based on limited data, okay? And so unfortunately, there's a bit of a crossover basically when it comes from the discovery setting. In vitro area, lots of data, easy to label. You ask a simpler question. If we're in efficacy or safety space, we have less data. It's very conditional data, right? But it's more in vivo relevant, right? And that's one of the problems right now when it comes to using computational systems and modeling data. Quite often there's a focus on the areas where we have data, but that's like looking for the key where the light is in a way, right? So you model the data you have and not the data you should model. So basically we need to move towards in vivo relevance of the data we model, right? So organ-based risk modeling, for example, combining PK with particular mechanistic endpoint models, that might be one of the ways for the future. But just to point out, data is not data, okay? There's very different types of data. In vitro, in vivo might be one way to think about it. So always look at whether we can label our data, how much data do we have, and is it in vivo relevant, okay? That's very important to assess when you look into using data for machine learning. Yeah, that's what I just outlined, right? There's a conceptual problem. So machine learning models which have an output variable, they are supervised models. You need to label your data, but assigning that label, the output label, to in vivo relevant data is actually very difficult. Things depend. They depend on dose and so on. Yeah, and that is a problem, conceptual problem, when it comes to machine learning in our area. But there's actually still areas where machine learning models based on labels can succeed. Yeah, I'll give some examples or one example here. But just to make that point clear, what labeling data means. Labeling data means that we say a compound is toxic. A compound binds to a particular target. A compound treats a particular disease. And then we can group compounds in particular toxic versus non-toxic classes and train a machine learning model. But this is a kind of the world is flat view, right? Because it means we believe our labels. Why is that naive? Because a compound is not toxic. That depends on dose, on exposure, right? Does a compound bind to a target? Well, in the disease context, depends on whether it reaches the disease tissue, right? And only then it's relevant that it binds to the target. Conditional again. Does a compound treat a disease? Well, depends on endotype of the disease, right? So basically, we need to label our data for machine learning, but in many cases that oversimplifies the problem. We can still use that concept in order to build machine learning models. Where can we do that? For example, to predict bioactivity of compounds. So that's something we did years ago with AstraZeneca. There's many approaches around. The concept is quite simple. We use bioactivity data on a large scale. And also in the public domain, Campbell, PubChem, there's data on the order of millions of compounds available, which we can use. And what we do is we build a machine learning model based on a molecule. circular fingerprints. I will outline that in a few slides. Circular fingerprints that are used to encode those molecules. And then we train a random forest. We'll outline that in a few slides as well, in order to predict bioactivity against the target. Why does that actually work in practice and is useful? Well, because the data comes from that in vitro space, right? It comes from high throughput screens, from biochemical assays, or at least simple assay systems that is not as conditional as in vivo data would be, which means in those cases, we can assign an IC50 to a molecule, bioactivity, okay? And that is actually data where we have lots of data available and is largely unconditional. In some sense, that is the two criteria that are really key in order to use machine learning in the life sciences. We need data A, which we can label. We need it on a large scale. And in order to be able to label the data, it needs to be relatively homogeneous. And that's the case here, okay? And these are generally the criteria. If you're on the other side of the table, when we have too little data, or we have data that is too heterogeneous, derived in very different conditions, for example, then it's very difficult to put that into a machine learning model. But keep in mind, and that's exactly what I actually illustrated before, but keep in mind that those models only cover known chemical space. So we only have in the model what we measure, right? That goes back to that data-driven question, okay? We only model what is in the model. And that actually means there's an asymmetry in the predictions. If we predict a certain bioactivity against the target, we actually don't have only two classes, active and inactive. We have three classes, because we might have evidence for activity, we might have evidence for inactivity, or we might have absence of evidence for either of those classes, right? And that means in practice, we actually have three classes, but quite often if you have computational predictive models, you actually lump the absence of activity class into the absence of inactivity, presence of inactivity class. So you basically say, if you don't have any evidence for activity, we say that the molecule is inactive, okay? So that means often predictions are not symmetric, is my point, right? And the unknown parts of chemical space, they are sometimes lumped together into the inactivity class in this case, okay? So predictions are not symmetric. That's often important to keep in mind as well. Okay, so in some cases, we can model life sciences data in the areas where we have enough data, where we can label our data, okay? But of course, especially if you come from the safety area, you're aware that that often isn't the case, and it oversimplifies the problem. We don't only have an IC50 against a particular target, right? If we have receptors, we might have partial or full agonists. We might have different residence times, bias signaling that's related to downstream side effects, for example, right? How do we actually, what do we call a disease, right? Is there actually, if you think about depression or we think about anxiety, these are not biologically defined diseases, they are symptom clusters, right? And so what do those examples illustrate? They illustrate that when it comes to in vivo data, it's often difficult to label data, okay? We don't know all of the aspects of compound activity. And even if we knew it, yeah, it would be a very conditional system that is difficult to encode in a machine learning model where we need an output label, yeah? And if that label is conditional, yeah, then we don't have large data sets that we can associate with the label, so we cannot train a model, okay? And that is actually one of the conundrums, one of the problems in machine learning right now. We have very powerful algorithms, but when it comes to in vivo relevant data, it's difficult to label the data that we have, okay? And there's then, of course, two workarounds, right? We can take the conditionality into account, or we say, okay, we label our data, and then we ask the model to predict endpoints. And afterwards, after the prediction, we take into account what that prediction in the given context actually means, right? That's the two workarounds here, okay? But the point here is we have very powerful algorithms. And if you hear about AI, that has made great progress, right? Image recognition, speech recognition, and so on. Due to this labeling problem, it doesn't really translate to the drug discovery area as easily, right, as to other areas. In other areas, you don't have that conditionality in the data. And one example of that, sometimes you see in machine learning models, machine learning journals, you see a classification model, does a drug cause an adverse reaction? And then it's a binary classification model. Is that actually a relevant model you can use? Well, it actually oversimplifies the problem, right? In which cases do you see an adverse drug reaction of a drug? Well, depends on the patient genotype, disease endotype, then it certainly depends on the dose, right? And then reporting systems are very, are not ideal, very biased as well, right? FAIR's FDA adverse event reporting system and so on. It's very biased data sets. quite often you don't have a binary classification. Does a drug cause an adverse drug reaction? It depends on the sex, and then you have different severity, and then you see an adverse reaction in 20 or 50% of the cases. Those numbers are very difficult to get as well. Maybe you see the side effect only after repeat dosing, maybe only in males, and so on. Right? This is what the real world looks like, right? So labeling in vivo data is very, very difficult. You know? And this is to illustrate the difference between the areas where computer science has made great progress. Machine learning can be really applied in image recognition, for example. So here we see an image of a cat. That cat is represented as a matrix, and that matrix goes into what's called a convolutional network. Convolutional networks detect shapes, right? For example, the shape of an eye and of an ear and so on. And then you have output layers that says, I have 2 eyes and ears of that shape, and that means there's a cat in the picture, okay? And so there's a smooth integration of the image representation as a matrix, because that's how you represent an image in the computer, right? You have RGB values in that matrix. Then you have a convolutional network that operates on matrix, very good integration of information. And then you have an object label that's largely unconditional. In drug discovery, that's different, unfortunately. You have chemistry. That chemistry can be, or that molecule here can be protonated, might be reactive, you might have different crystal forms, and so on, right? Then there's the question, how do you actually describe that to the computer? Trial and error, right? Log P, different functional groups, and so on, question mark. How do you encode that in a neural network, for example? Well, trial and error again. And then the output label is conditional, okay? And that illustrates in the image domain, you have quite a smooth integration of information going from an object to the representation, to the modeling method, to the label. In the chemical domain, you don't really have that, you know? Your molecule is alive, so to speak, right? There's not only one form, right? Conformations, protonation states, and so on. You can represent it in different ways using different descriptors and so on. So that pipeline isn't as smoothly integrated, okay? And that's one of the problems of translating approaches from the computer science, from the image recognition area to the biological and chemical domain. There's also a few problems with the data that are due to the pipelines we established in the last decades, of course. Since the 80s, throughput screening came up, right? So there was the need to profile many compounds quickly and cheaply. And that means there's quite a lot of proxy data around. Let's say the glue-gel mitotoxicity assay you might be familiar with, right? So what does that data actually mean? We have, it's quite a noisy assay anyway, and you take a ratio of activities, so it's difficult to deal with the data in practice. But what does it mean conceptually? We have mitochondrial toxicity, so a certain safety endpoint, and that data gets measured in a proxy assay that represents one aspect of the mechanisms that contribute to mitotoxicity, right? And if you then use AI models, machine learning models, you actually build another model on top of that. So you have a models of models situation, right? And that means that quite often the machine learning models we have in the safety space, they're actually quite far away from the in vivo situation that we're trying to predict for. And that's, of course, true in other areas as well. We say inhibition for DILI, for example, and so on. So in many areas, we don't really have the right data that we meant to model in order to predict in vivo relevant endpoints because of that models of models situation. And this is the way I think we should think about data. So we need to ask the question, what is actually the question we want to answer? What's the data to generate? How do we represent our information? And then comes the machine learning on top. Okay, goes back to what I said in the beginning. Data comes first, then the representation, and then the method, right? Right now, there's quite a lot of attention on the machine learning methods, deep learning, and there's more layers in my network than yours and so on. But actually, that's all based on the representation of information, the data I have available, and the question or hypothesis. So is it up or down regulation of that gene, right? Does that give me a signal? Do I need to look for particular reactive functional groups in my molecule, right? And that tells me whether I have the right data and the right representation, basically to build a machine learning model. Okay. Yeah, let's go to models. So descriptors. How can I actually describe molecules? So let's go back to that slide again. So experiment, molecule, we measure an endpoint. And if we have a machine learning model, we need to encode the molecule using a descriptor. And then we have the machine learning model on top to predict an endpoint. How can we encode a molecule in the computer? That's by and large trial and error. And there's different types of descriptors, one-dimensional, two-dimensional, three-dimensional. That's a common way to characterize them. What do we want to do with a descriptor? We want to have an information-preserving representation of data as an input for the model. And that it sounds easy, but actually it's quite difficult to achieve because in many cases, we don't really know which aspects of the molecule are related to the endpoint we are interested in. In some cases, I know, okay, it's reactivity, it's log P, it's molecular weight, but often I don't really know that. And there's different ways to encode a molecule. We can stick closer to the connectivity table. We can use shape-based descriptors and so on. I can do nearly anything I want. There's a book by Toshini about molecular descriptors that lists 7,000 or so molecular descriptors. Basically, you can come up with any descriptor that works. That's a short answer. And you can classify them in different categories. And here are some examples. At the top, there's a circular fingerprint, as it's shown. That's based on the connectivity table. I'll go back to that in a slide or two. At the bottom left, you see a pharmacophore-based descriptor. So you have feature points in 3D. Yeah, so that depends on the conformation. The two-dimensional descriptor doesn't. And at the bottom right, you have a surface charge-based descriptor. Okay, so it does not take into account explicitly the connectivity table. It's also not pharmacophore-based. It takes charges on the surface into account. Yeah? And if you model an endpoint, you always need to see, how do I encode my molecule for the computer in order to then build a machine learning model? So basically, every feature you get from one of those descriptors is one feature, and then the machine learning model fits the coefficients in the front of that. That's the way to think about it, okay? Every feature is one variable in your model, yeah, but that can be used in order to describe correlations between input and output space. But if your variables don't make sense, yeah, then you cannot fit a proper model to that either, okay? And that's here displayed on the right. So that's a fingerprint, presence and absence of variables. And those variables need to have some meaning in the given context of the model. And context can be bioactivity, log D, or some safety endpoint, but these are the variables you feed into your model, basically. Yeah, to give you a few ideas of what descriptors can mean, there can be one-dimensional descriptors. That's one of the standard ways to classify them. So it's basically a number you assign to the molecule. Molecular weight, molar refractivity, so that's a shape-based descriptor, or log P, and so on. So in those cases, you derive a single number from the whole molecule. They can make sense, in my opinion, only if there's a relationship between, in the case of physical chemical properties, basically. Yeah, log D, molecular weight, maybe some shape-based descriptors, moments of inertia or so. Do you have a long rod, basically, or do you have something more spherical? In those cases, one-dimensional descriptors can make sense, in my opinion. There's also so-called topological indices around that are then more graph-based descriptors, but in practice, they don't really matter. So for one-dimensional descriptors, I think it's only phys-chem-related properties that make sense to describe a molecule. And then you have descriptors that are based on the connectivity table of the molecule. So here's a descriptor that's called the circular fingerprint. Why is it called circular fingerprint? Because you look at a heavy atom you put into the center of that descriptor. That's an aromatic carbon in this case. That's the at layer 0, as you call it. So that's the central atom. In the next layer, you have another 3 aromatic carbons, right? These are 2 aromatic carbons, sorry, 1 sp2 hybridized carbon. And then the next layer, you have another set of aromatic carbons. Here you have your amine and you have your carboxylic acid, right? And so these are the atoms that are in the next layer of that circular fingerprint. And you do that for every heavy atom in the molecule. So this example is for this particular carbon, but you also calculate that descriptor for this carbon, that carbon, that carbon, and so on. And this way, you actually have a descriptor, and that isn't a descriptor that works very well when it comes to bioactivity prediction, for example. That captures quite a lot of the bioactivity-related features of the molecule. So by trial and error, and that's actually something I did in my PhD a long time ago, that circular fingerprint descriptor is actually a very good descriptor to capture molecules that might be even substructurally different, have a different scaffold, but that are active against the same target. Okay, so empirically, it has been shown that this descriptor actually is a descriptor that contains lots of information related to bioactivity. So that's what you can use to build a machine learning model for bioactivity-related endpoints. So that's one of the variables that you have in your model then. And there's also three-dimensional descriptors. So no, yeah, three-dimensional descriptors. And here are pharmacophoric point descriptors, for example. So you can say you assign a property to every atom in your molecule, and that may be individual atoms, it might be groups of atoms. You have a benzene ring here, for example, so you can say that I treat that as a lipophilic feature, so that's why it says an L here. And then I have, for example, the oxygen on top here of the carboxylic acid, right? That's a hydrogen bond acceptor. And then I have the other oxygens here. So you have a carboxylic acid, so that would be deprotonated at the bottom, right? So you would probably assign a negative charge here. That hasn't been done here because the molecule is not deprotonated, but in practice, of course, you would assign a negative charge here, right? And then you can say, which types of properties do I have? Lipophilic feature, hydrogen bond acceptor, carboxylic acid, so negative charge. Then I can say, what are the distances between those points? And that's then what I would call a 3-point pharmacophore, right? Or 4-point or 2-point pharmacophore, depending on how many points I take into account. Yeah. So that means I do two things. I have my molecule in 3D space, so it's conformationally dependent, that descriptor, and I assign properties, and then I take the geometry across those properties into account. And so we went through 1D, 2D, 3D descriptors, right? 1D single values make only sense, in my opinion, in case of Fuscam properties. 2D descriptors, connectivity table based, independent of conformation. 3D descriptors, for example, pharmacophoric triplets, right? And these are the features you can put into a machine learning model. The point of this plot is only, there are lots of abbreviations here, is all descriptors behave differently. So what do you need as input into a model? It depends. So first of all, it's better to understand what you're actually trying to model and to put some information in choosing the right descriptor. But after that, it's trial and error, right? Think about a read across, for example, if you come from the safety area, right? You're trying to group molecules that belong to the same series in some way, right? And you do that by looking at some scaffold or some property space, for example, yeah, in order to group molecules in a suitable way. But beyond that, it's then basically trial and error if you want to predict molecular endpoints. Okay. The point of this slide here is to show you can derive features from the chemical structure. Okay, 1D, 2D, 3D. But you can also use biological response as a descriptor of molecules. This is what Srijit, who will present later, for example, did. There are cell painting, cell morphology endpoints that are being used right now by the Broad Institute. There's also a HESI initiative to use cell painting descriptors in order to anticipate organ toxicity. Right? And here the question is, how can I actually use the biological response of a system as a descriptor? Because sometimes it's not in the structure. We don't really know what that means for the biological system. So if I put that molecule into a biological system, a cell, I do a cell morphology readout, so cell microscopy, basically. I stain particular compartments and so on. Then I do microscopy. Then I use that biological response, changes in geometry as a descriptor, right? That's what you can also do. And the point here is that sometimes we don't know what matters of the chemistry. We need to ask the biological system to give us a response, right? And that can also be a descriptor. That, for example, groups certain types of toxicity and so on. And this is what Srijit is working on. Yeah, so you can derive descriptors from chemical structure or use biological response. Can be cell morphology, can be gene expression, of course, as well. Yeah, can be many types of biological response. Okay, and now we actually have the first part solved, right? So we have a descriptor that can be 1D, 2D, 3D, can be biological response. And then afterwards, we need to fit a model, okay? So we need to use the variables that the descriptors provide us and fit the coefficients in front of those variables, basically. That's all the model does, right? So you train your model and then you test it. So training means fitting your model. Testing means you use that model to predict endpoints for a novel data set. And so there's different types of models, but they usually fall into one of those three categories. We can do nearest neighbor methods, so one-to-one comparison. We can do clustering-based methods, would be an unsupervised method, but I can put in landmark compounds where I know what they do. And if a new compound clusters with that compound, I can extrapolate activity. Or I have machine learning models that's shown here at the bottom. What do those machine learning models do? Well, they look at all the information of the different descriptors. So that's what those white and black boxes mean here, presence and absence of features, right? And then they scale, okay, the evidence in the whole data set. In 80% of cases, I see a certain feature, in 20% I don't, right? And so you weigh the different features in your final machine learning model. That's all a machine learning model does, yeah? It scales features, yeah? It fits coefficients in front of the features in your particular data set. And so all of those machine learning models, they have different underlying assumptions. Linear regression, for example, right? You assume you have a linear relationship. If you have something like a k-nearest neighbor method that's shown here, you assume that the neighborhood defines behavior of a particular molecule. If you have a decision tree, so this is something I'll outline in a second as well, you actually have an assumption that a series of decisions of different features, log P is higher than 5, molecular weight is lower than 300, then your compound gets into the brain. So that's a decision tree structure. You have a series of particular features that actually decide class membership, okay? All of those models have underlying assumptions of some sort, yeah? Linear relationship, different types of relationships, combination of features, and so on. That's all what the difference between machine learning models means. It means that we have a different assumption of how to combine features and whether those combinations should be linear or nonlinear. Yeah, that's all that the machine learning model does. There's one thing that's very important in machine learning. We want to have a good model, high classification accuracy, high correlation coefficient, and so on. But future applications are on new data. And that's a bit of a schizophrenic situation because we want to have good model performance, but quite often, let's assume we have a model based on two chemical series. One is toxic, one is non-toxic. Okay, we will have a perfect model because the series look very different. But what does that mean for the applicability of my model to new molecules? Well, it's useless, right? The model knows only about two chemical series, doesn't learn about any underlying relationships. So what does that tell us? We need to have a trade-off, right, between model performance and the ability of the model to generalize, right? And usually ability to generalize, to make predictions for novel data is what matters in practice. So don't only look at model performance, yeah? That can be very misleading. Like in the example I've just given, you need to see if the model generalizes based on new data. And the particular modeling methods that we will use here in this practical in a few minutes, they will be decision trees. So what do they look like? Well, I have a series of decisions that use, for example, physical-chemical properties to discriminate groups of compounds. So on the left-hand side, there's a generic decision tree. So I put in all compounds at the top, and then my decision tree machine learning method tries to find the first feature that gives us the clearest separation of groups of compounds. Blood-brain barrier permeation is here one example, right? So the model goes through total polar surface area, log-D, molecular weight, and so on, as is shown on the right-hand side here, and sees what is the variable that gives me best separation between BBB permeable compounds and non-permeable compounds, right? And then I see I have a different distribution of BBB permeable, non-permeable compounds in the next branch. Then I make the same decision again. I go through all of my variables and see what is the variable that gives me the best separation again. Okay, and that's what the machine learning model does, what a random forest does, or a decision tree in this case. Random forest is then multiple decision trees next to each other. So you're looking for features that give me clean classes, basically. And that's what we did here on the right-hand side, for example, for BBB permeation, efflux, substrate likeness, and so on. And what you see is that you have particular ranges of total polar surface area, as well as the log-D or molecular weight that really then determine whether compounds get into the brain or not, right? So that's the decision tree structure. Yeah, you're looking for features that give you pure classes. We define pure classes and so on. There's many different ways to do that, but that's the concept. Give me the variable that gives me the purest classes in the next layer. And that's what you repeat, and that's your whole decision tree, right? In this particular practical, I think a random forest will be used. And here you see a random forest. Well, one tree is a tree, right? Many trees can be a forest. So that's the same concept here. So we have multiple decision trees that are trained using slightly different variables. Yeah, and then they give you slightly different predictions. And then afterwards, you use the output of that decision tree, following the sequence of decisions, right? Is my blood-brain, is my molecular weight higher than a certain value? Is my log-P higher than a certain value, and so on? And then you have a leaf node, as it's called, the bottom node, that gives you the class membership that you predict. And you basically use a random forest, so a combination of decision trees, to do a majority vote. So two of the trees say one thing, one says the other, then you take the majority vote of those two trees to predict whether a molecule gets into the brain or not. Deep neural networks work slightly differently, and this is shown here. The concept is very easy to understand. In practice, sometimes difficult to train models, but the concept is very easy to understand. So you're putting in your variables on the left, so molecular weight, 300, log-P of 3, and whatever, that's what you put in on the left. And then you have the hidden layer nodes that actually take the input. So the numbers you basically put into those orange nodes here, and they assign a weight as well as a shift function to those input values. Okay, and that's what gets done multiple times, and then you calculate the value of the output node. Yeah, and so what do those nodes precisely do in the hidden layers? Well, that's shown here in this particular figure. So on the left-hand side, you put in the input values, molecular weight, log-P, and so on, and then you see this logistic function here at the bottom, right? So these are the input values here, the x's, the x-axis, so the different x values. So these are the descriptors you put into a model. Okay, we had that before, molecular weight, log-P, or whatever you want to put in, right? Then you have different weight values. You sum over all of those products, and then you have a different shift, right? And that is the output of that node. And that's the whole, if you understand that, you understand how a neural network works or deep learning works, right? So you have your input values here, and then you basically in the next node, you sum up using different weights, the input into that node. And then you have a logistic function. You need that in order to model nonlinear spaces. You have a logistic output function, activation function, and that basically gives you the output of that particular node. That's all a neural network does. You know, if you understand that, you understand deep learning. Basically, you do that many, many times. You have many parallel nodes. You have many layers, one after the other. It's practically not easy to train. I have to say, you need some tricks and so on. But the underlying principle is exactly like this. You take the input descriptors, assign weights, logistic function, and you forward the output. Okay, and you fit all of those parameters, the W parameters here, the weight parameters, in order to have the best predictions in the end. Okay, that is what is called model training, but that's the whole concept of a deep neural network. How do you evaluate that actually? So if I have a binary predictions, I have actual values. So these are is the ability of a compound to get into the brain or not, for example, and I have predicted values. Yeah, and then I have true and false predictions. I can have true negatives where 0 predictions agree. Zero prediction agrees with an experimental 0 value, or I can predict an 1 as an output, gets into the brain, for example, and the actual value is a 1 as well. So these are the true values, okay, in my confusion matrix, as it's called. And I can have wrong predictions, where I can predict wrong in both ways, right? I can either predict as 0 and the actual value is 1, or the other way around. And this is the confusion matrix that I can use to establish performance of my model. Yeah, the more I have in the green boxes, basically, the better my model, and the less I have in the red boxes, the better my model as well. Based on that, I won't go to detail here, but I can calculate different performance measures of my model, right? Accuracy and recall and precision and so on. They are all derived on that particular confusion matrix. There's 2 different types of performance metrics, and that is very important. One is based on the overall distribution of parameters. The other one is point-based. So correlation coefficient, for example, right? You can have an R² of 1. Yeah, you have a perfect correlation of your model, but that gives you a performance across all data points. And that can often be misleading. Why can it be misleading? Imagine you have a group of compounds that has very low toxicity and a group of compounds that has very high toxicity. Yeah, and you predict the group membership correctly. Yeah, but you do not differentiate between members within the group. Okay. Then your model is close to useless as well, because all the model does is it predicts which cluster your compound belongs to, right? But it doesn't discriminate within the clusters, okay? And basically, if you have cluster data, then correlation-based performance metrics can be very misleading, right? And you always need to look at the distribution of your data as well. If you have very clustered data, you can have a very high correlation coefficient. If you have just one cluster of compounds here and one here, you have a very high correlation coefficient. But if you cannot discriminate between compounds that are located within the clusters, the model is still useless for lead optimization and those things. So cluster data can be very misleading when it comes to correlation coefficients. So in practice, RMSE, root mean squared error, or mean standard error, and so on, are often better ways to determine the performance of your model. So you know, my prediction is 0.7 and the root mean squared error is 0.1 or so, right? That tells me what is the error of that data point. If I know only that my R² overall is 0.8, for example, it doesn't tell me anything about what the error of a particular data point is, okay? So look into correlation-based versus point-based error metrics. Point-based error metrics are usually what you want. It's always also very important to look into the distributions of your data points. Here at the bottom, you see different distributions of data points, predicted versus experimental, for example. They're all of the same statistics, mean, standard deviation, and so on, okay? But the distributions are very different, okay? And some of them could be used for models at the top left, for example, but some are close to use less at the bottom right. So always look into the data, always look into the core data. Don't believe overall statistics of models. So if someone gives you a prediction for a particular data point, ask for the predictions for the test set and look at the distribution, because otherwise that can be very misleading. Statistics doesn't tell you the full story. There's a saying, on average, two people are walking over a bridge, but actually one is flying, one is drowning, right? The average doesn't tell you anything. Look at the individual data points. You always need 2 numbers to characterize an output: an experiment mean and standard deviation, in a model predicted value and, for example, confidence or error. you always need multiple numbers to characterize any distribution. So always ask for the confidence of the model along with the predicted value, or applicability domain, or the error of the prediction. It can be different names for that. There's quite a number of problems with chemical data biases, for example. There's lots of focus on explainable AI right now. It can be actually assigned the meaning of a particular, it can be actually understand why the model does a particular prediction. And this is an example here of my own work, so I'm very happy to point that out as well. We were trying to find out which chemical features are actually related with bitterness of compounds, okay? Chemical space is large, 10 to the 60 compounds or so. Our data is often very clustered, okay? Fast follower compounds in pharma, for example. If you have published literature, you have series and so on. We never have even distributions in chemical space. We don't even know what that is in some sense because we cannot define it. So in this case, we were trying to classify bitter compounds versus non-bitter compounds. What did we find? Well, sugar rings were characteristic features for bitter compounds. Why was that? Natural products are often bitter. Natural products are often glycosylated, so contain sugar rings. And that means it's a confounding factor, right? And that's why as characteristic features for bitter compounds, you find sugar rings. You have some sugars that taste bitter as well, but that wasn't the reason here. It's a confounding factor, right? And that's often very misleading in chemical data sets. Be careful with biases in your data when it comes to chemical data. Yeah, validation. So why is validation actually, what does validation mean? Why is it tricky? So validation means that we have a training data set. We want to validate the model and predict for the test set. Yeah, so that's an external data set that gives us an estimate how good behavior, how good performance of our model is. But the problem is always, how does that extrapolate to novel chemical space? And that is difficult to do in practice. So here's a way where people try to approximate that. Cross-validation is called. So you have a training data set to train your model, and then you're trying to predict data points outside of the training data set, and that gives you an estimate outside of the training data set, so for the test data set, that gives you an estimate of how well your model performs on novel data. And you can repeat that multiple times. So here's five-fold cross-validation, as it's called, right? In order to get an estimate of how well your model performs on novel data. But that's tricky to do because in chemical space, it's different from biological space. In biological space, we have different transition likelihoods. So mutations, for example, that can occur. So we can say, how likely is it my glycine actually mutates to an alanine? Yeah. So we have transition likelihoods. We don't have any of that in chemical space. We have very clustered data. We don't know what chemical space looks like, which means we don't know what biases are. We cannot do proper statistics and so on. Yeah. So in practice, so the previous slide was very idealized. So in practice, we have a training data set, a validation data set. This external test set to estimate performance in many cases overlaps with the data we had before. So we just take it apart, right? We set one part of the data apart for validating our data set into the test set. But the problem is it's drawn from the same data set in the beginning, right? And so that means that's a very biased estimate of performance. And then the next project we apply our model to can be in a completely different area of chemical space, yeah, where chemical modifications mean something completely different. Yeah, and that is one of the big problems of predictive models in chemical space. Chemical space is large. We don't know what biases are, how biased it is. Yeah, we cannot characterize it. And performance of a model based on chemical features is different in every area of chemical space. Yeah, so that makes it so difficult to estimate performance. Okay. Another problem is that we actually have two different parts we want to validate. And one of them is actually model validation, the other one process validation. And that's an important difference when it comes to machine learning in some other areas. And drug discovery, model validation only means we have data, we predict endpoints, and we say how good our predictive model is on that data set using class averaged accuracy or so as a performance measure. But drug discovery is much more than that. You have a project context, and it's something very different if you work on a late-stage cancer versus a lifestyle disease. Predictions depend on compound PK, right? If I predict mitochondrial toxicity with my model, well, it depends on PK, depends on the dose in man, depends on many things, right? So that is one thing that is neglected when I only look at machine learning models. The other one is follow-up assays. I was working at Astra, Computational Safety Group, for a while, and there the question was not, how can we make the area under the curve slightly better? Here the question was always, okay, if we predict something, what comes next, right? So what's the follow-up assay? Is it easy to run? What does it cost? How long does it take? And so on. So there's a disconnect between those things, and we need to care about the overall process, not only machine learning models, but embedding in the overall process. Right? There's two resources, review articles. They're on the slides because I'm running out of time here a little bit. So in case you're interested in that, please look them up. They're on the slides I was just posting to the chat. And finally, a few predictions related to predictions, a few questions to ask your friend, the modeler, based on what I've shown before. So the idea is we have a computational model that makes predictions, right, for novel molecules. The assumption is that the rules learned during training apply during model application on new data points. How can we gain confidence? So basically, why should I trust the modeler that the predictions are worth anything, right? And here are a few questions you can ask. So key goal is to find out how good is the prediction from a new compound. A few questions to ask is, what's the number of data points in the model? And is the chemical space coverage relevant for my application, right? Do I work on macrocyclic space? Do I work on natural product space? Do I work on GPCR ligands? Do I work on different chemical space? Do I have the right types of data in my model? Another question I can ask is, what's the nearest neighbor? And is it relevant given the particular question being asked? Think about series read across, for example, as well, right? So do I have a compound that belongs to the same series? Yeah. Do I know something about the binding pocket in the receptor? Do I get a clash if I extend that chain? Yeah. So is it relevant given the particular question being asked? Yeah? That's a good question I would ask the modeler as well. Related to descriptors, how was the descriptor chosen? And is there a mechanistic rationale for the choice? Yeah. Do I need to have, if I have a warhead, for example, in a compound, right? Do I need to have that warhead? Does my descriptor know about that? If I don't insist on having that particular interaction with a metal or something in the binding site, right? The prediction is worthless, right? Because it doesn't understand that there's a particular key aspect of the structure that I need to focus on, right? So do I have the right descriptors in my particular model? Yeah? Is it generic similarity? Often not so good. Yeah. Is it something that is related to my problem at hand? Related to models, was there an external test set used in the model validation? Was it large, diverse, relevant to new compound predictions? Does not model performance actually change depending on parameter choices and training test set splits? Yeah. If I have a data set and every time I do this cross-validation, my output is different, well, then I cannot really trust the model when it comes to new predictions either. Is there an applicability domain or confidence that the model assigns, right? Does it actually work on the external test set? Yeah, and rather often it does not. So a model performs very well in cross-validation because it's the same chemical space. So apply to something new, it doesn't, right? So basically, it doesn't my model extrapolate to novel data. And if all of that is answered satisfactorily, then I have suitable data in my model, I have a suitable descriptor, right? And I provide a confidence in the prediction. So that's basically as good as it can be. Yeah, so to summarize, so computational models are probably generally helpful to find patterns in data sets. Also in safety, I described limitations of data, yeah, in vitro versus in vivo, very important. Yeah, do I actually have data of the type I want to model? How do I choose descriptors, right? 1D, 2D, 3D, and so on. Validation, I have this cross-validation, but chemical space is large. So what does it mean for future predictions? And there's limitations of data when generating models from input space and output space. Chemical space coverage, right? Output space is, do I actually have, it can actually label the particular data that I have in my model. And I was outlining some general guidelines to ask your friend, the modeler, yeah, in order to increase the confidence one can put into the models when predictions have been provided. Okay, there's 2 or 3 resources here as well. So there's some articles in drug discovery today that outline the challenges when using life science data in drug discovery, artificial intelligence in drug discovery, what is realistic, what are illusions. I can recommend give those a read. There's also an article on how to lie with computational predictive models in drug discovery. It's not on how to lie, of course. It's about how to actually spot if something is not as accurate as it should be, right? And to prevent you, if you build a model, to actually prevent you from inadvertently lying as well. So in order to achieve a model that's as useful in practice as it can be. Okay, great. Thanks for listening. If there's time for any questions, very happy to go through those as well.
Thanks, Andreas. I think that was a great introductory section for those of us who are not too familiar with the way of artificial intelligence and how that can be applied to some of these small molecule data sets that we're perhaps familiar with. Yeah, does anyone have any questions? I've seen 1 or 2 pop up in the chat box. The also aim of the next session is we'll probably start in about 5 or 10 minutes with Layla's first exercise. But in the meantime, if you can continue to put your questions in the chat box, and between us, we'll do our best to answer those.
Yeah, there's actually one here, right? If there are movements in toxicology to compile data from companies to create a large quality interoperable database. I mean, what I can see is there's, for example, the IMI and related initiatives, or Hazy, for example, is doing that as well, right? Consortia are being formed. Much of the data in pharma is, well, confidential data, and even with pre-competitive data, so related to safety and so on, there's often reluctance to really make that data public. So there is some consortia like the IMI consortia, eTox, eTranssafe, and related consortia, right? They try to compile that data. However, that data has been compiled historically for different purposes, and you need experimental design, in my opinion, to really have useful chemical space, endpoint space coverage. So that is not ideal data. It's also quite often data that hasn't been annotated as well as it should be to be really useful. So probably we need to do something more prospectively to drive the field forward. Pharma is different from many other areas. You mentioned your Gaia X. So pharma is different from many other areas. So there's generally quite a reluctance, unfortunately, to share data. But we need more consortia, especially with experimental design, to have the right chemical space in the data sets that we are going to generate. Okay, yeah, I think, no, thanks for listening then, everyone. I think I'll hand over then to Nic as well as Layla and Srijit for the more detailed hands-on tutorials. Thank you.
Thanks, Andreas. Yeah.
Right. Hi, everyone. I'll start sharing my screen. Can you see that okay and hear me okay?
I can certainly see that.
Yeah, all good?
Yeah. So what I'll do is everyone into the chat box. I'm now pasting a URL link that should take you to the Google collaborative environment that Layla is working in currently. Any issues, then please just paste into the chat box, and that link should take you straight to this page that we have open here in front of us.
Yeah, and what you're going to want to do as well is, because you can't currently edit this file, so if you want to kind of play around with the code, you're going to need to save a copy of your own in the drive. So if you click File, Save a Copy in Drive, it should open up a new tab, which has made a copy of the code and is called Copy of in Silico Toxicology Hands-on Workshop. So that is what you're going to be working on. So yeah, if you could make sure that's all okay, and then yeah, if it's not, put it on the chat with the technical hashtag and we'll be able to help you out. So should I give maybe a minute or so just for people to get started on that? In the meanwhile, I can kind of introduce the workshop. So yeah, welcome to the hands-on workshop part of the session. I'm Layla, and Srijit is going to be taking over and doing a part 2. So yeah, as Andreas alluded, safety modeling is very tricky. It's actually something we're doing at AbsoluteAI. So here we're going to take you through kind of simple like first steps for building machine learning models for safety in Python. So we're going to be using in vitro data sets in this case. So I'm going to be taking you through modeling hERG inhibition using a regression model. And then afterwards, Srijit is going to take you through modeling mitochondrial toxicity using a classification model. So I'm also going to give a bit of a kind of overview or rundown or introduction to Python and a few of the specific libraries that we're going to be using, as well as a bit of a rundown on Google Colab itself. So if everyone's all right, I guess we can get started now. So the first thing you need to do for this workshop is you need to get the data and also install a few packages. So in the Colab notebook, you'll see a block here that has some code in it that says you want to install a few packages, and it's also going to be downloading a few files. So if we click that play button, then it should start to kick in and start installing things. So while that's going, I can talk to you a bit. Oh, it's just connecting. Give me a second. Initializing. Okay, connected. Okay, so yeah, clicking this, as I said, installs a few packages and also downloads the data sets that we're going to be using. So now I'll give a bit of a background now to Google Colab notebooks. So Google Colab notebooks are great because you can put in blocks of code and text and images all in the same document, which is this one here. You can share it, which we've done as well. And we can run Python directly from our browser, and it's actually using compute power from Google. So you saw here in the corner, it was connecting. It's actually connecting to compute power from Google, which is what we're using to run everything today. So this page is interactive, and if you've saved a copy in your drive, then you'll be able to edit it. So each block, so you can see that if you click on kind of different sections, each one of these is called a cell. So cells can contain text. So if we double click on a cell, you can see that this is a text cell. They use a simple language called Markdown, where you can use to make bullet point lists, italicize text, or make it bold. So code blocks, on the other hand, they use, this is Python code. So you type your code in, and when you execute it, the code is executed. So you can either execute a cell by pressing play, or you can press Shift and Enter. So for example, this cell code here is going to add up a series of numbers and print the result. So here we're saying x is equal to 1 + 2 + 3, and we're returning x, which is 6. If we want to add new blocks to the notebook, we hover where we want the new cell to be. So if I hover under here, you can see a line comes up that says plus code or plus text. So you just click on it, and then you can edit your cells there. So now I'm going to give you an introduction to Python. So Python is the most popular language for performing machine learning. This is because the syntax is quite simple compared to other programming languages, and there are many open source packages for chemoinformatics analysis and machine learning itself. So to take you through some of the basics, so we're going to go through some simple operations in Python. So in Python code, if you want to kind of comment, which is something that's usually quite useful to do if you want to make note and kind of document what your code is doing, you just put a hash symbol, and then everything you write after that isn't run as part of the code. So you can see here in this code cell, I've got hashtag and then some operations, brackets, this isn't run. And when we run it, this isn't executed as part of the code. So the print function just prints the results. So here we're printing how many times does 3 go into 10? If we divide 12 by 5, what's the remainder? And then what's 4 to the power of 2? So if you press play, you can see that it prints each of the results of these operations. So in Python, what's very useful to do is to define what's called a variable. So a variable is just a way of storing a value. So here in this next code cell, we have x = 5. So x is a variable, and we can perform different functions on this variable. So here we've said x plus = 2, which is the same as saying x is equal to x plus 2. And then if we print x, we see that it's added 5 plus 2. So just to check that everything's working fine, can I ask you all to kind of create another code block, which instead of doing this, instead takes 3 from 7. And I'll give you a minute or so to do that. And then if there are any issues or the code isn't working, you're not able to edit anything or run anything, then yeah, please do write in the chat, and we'll hopefully be able to help you. So yeah, just to go over again, the hashtag means that it's not run, it's a comment, it's not a run as part of the code. We can print things, we can run mathematical operations in Python, we can assign things as variables and then edit the variables and do things to them here. So yeah. So I'm going to just wait another couple of seconds and just make sure that no one's having any issues, and then I'll carry on. Okay, I think that means everyone's okay. So as I just mentioned, variables are used to store information to be referenced and manipulated in a computer program. So in this above code, as I said, X is an example of a variable. We can also store collections of data as lists or arrays. So lists can store strings, so text or values such as floats or integers, and also mixtures of different data types. Whereas arrays store values, and then these can be created using a package in Python called NumPy. So if we want to use a package in Python, we need to import it. So we're importing NumPy here, and then we're actually calling it by its alias, which is NP. So we're importing the package, and we're saying that we're importing it as NP. So once we import the NumPy package, we can now use the functions that are within it. So now I'm going to show you what I mean by a list and an array. So to make a list, all you need to do is create a set of square brackets, and then within the bracket, you have each element that you want to put in your list separated by a comma. To form an array, you do something that's very similar. So again, you have a square brackets with your numbers separated by commas, but we're calling the function here np.array. And what this means is that we've imported NumPy as np. So it's using the functions from NumPy, and it's taking this array function from NumPy and it's applying it to this. And then what we can do is we can print the list, print the type, also print the array, and print the type of the array. So if we execute this block, when we print the list, this is this here. So we've just printed exactly what we've input here. The type of the list is a list, which is what we expect. Printing the array, you can see here, it looks slightly different. So when you print the array, it doesn't have the commas in between. And then when we print the type of the array, we see that the class is NumPy array. So if we want to get the length of a list, we use len. So if we put len and then open brackets and the list that we just created, we get 5. So that means that we have 5 elements in our list, which we can count 1, 2, 3, 4, 5. So that matches just fine. And then if we want to extract a particular element from our list, so say we want to have the first element from our list, we can subset using square brackets. So here we're saying we want the first element. Oh, did someone say something? Let me just check and make sure that everyone's okay. So to extract, so as I was saying, extract an element of a list, we put the index that we want to retrieve in the square brackets. In Python, it's 0 index, so 1 starts from 0. So if we want to get the first element, we need to get the 0 index. So if we play that, then you can see that it's extracted the first element from the list. So something that you can have a go at now is to try and extract the third element of the list. So the third element would have an index of 2. So when we print it, we can see that we've got the 1, 2, third element. So again, if you can make sure that you're running that okay and it's all running okay, and then put any questions in the box. And the way that it's working is that Srijit's keeping an eye on the technical questions, and then if there's anything that he thinks I need to kind of raise to the group, he's going to let me know, and I'll kind of say it out loud. So to go back, so go through what we just did. So we imported NumPy, which is a package which is used for various reasons, but what it's mainly used for is for manipulating arrays. We've created a list by just putting some elements in a square bracket. We've created an array by using the array function from NumPy, which we imported using its alias. We've printed the lists and the arrays that we've made, and we've verified that the types are what we expected, which is a list and a NumPy array. We've also managed to see how we can get the length of a list and how we can extract a particular element out from a list as well. So something else we can do in Python to save a lot of time is something called defining a function. So if we want to create a function which adds all even elements of an array, this is kind of, I'll break down, it might look a bit scary at first, but I'll break down exactly what's happening here in this function. So we want to create a function which can add all the even elements of an array. So to do that, we need to first define the function. So we put def and then the name of the function we want. So we're calling the function array add. And then what's in the bracket here is what will then go into the function. So array add is the name of the function, and what goes into the function is an array. Because we're adding things, we need to start with a sum of 0. And then what we're doing here is we're doing something called a for loop. Now, what a for loop does is it goes through each element of a list or an array. And then you can kind of perform an operation on each individual element of an array for every single element. So when we say for I in array, we're saying for every element that's in an array, we do something. So what are we doing? So for each element, we check if the element itself is even. And to do that, we use this function here, because this tells us if we could divide it by 2 if we have no remainders left over. So for each element, we say, is it even? So if we put the statement underneath what happens once that kind of statement is true and that's passed. So we say that sum, which is initially defined as 0, is equal to sum plus the element, which is even because it has passed this statement here. If it doesn't pass that, it reaches this, which is called else. So else just means if this isn't true, then this. So if this isn't true, then we're skipping, we're continuing, because we don't want to add it to our sum because we're only adding the even elements of an array. So just to go through that again, we're defining a function here. We're calling it array add. Our input to the function is an array. We define our initial sum as 0. We then declare a for loop, which goes through every element in array, where every element is called is assigned the variable I. We use an if statement to check whether the number is divisible by 2 and is therefore even. If that is correct, then we add it to the sum. If it's not, then we continue. And then at the end of the function, we return sum after performing all of these operations. So if you press the play button, this will then declare the function, and then we'll be able to use the function. And let's check if it works as we expect. So we want to print the result of our new function, array add. And then as you saw here, it takes an array as input. So we're going to put in 2 different arrays here. So one is this one that has 3, 4, 5, 6, and the second one has 2, 3, 5, 7, and 10. And if we press play, what's happening here is this array is going into this function, and this for loop is being evaluated for every single element in the list, and it's being added up if it's even. So another common data type that's used in Python is a dictionary, the shortened to dict. So these are denoted by curly brackets. So as you recall, lists were denoted by square brackets. So when we define a list, we use square brackets. Whereas when we want to define a dictionary, we use curly brackets. So dictionaries are a collection of key value pairs. So we map keys to associated values. So to give an example here, we have a dictionary which maps a location, which is the key, to its corresponding value, which here is its Major League Baseball team. And then how we define that, how we kind of put that into a dictionary is we first say, okay, well, we're making a dictionary, so we need the curly brackets around it. And then we just have our key, and then a colon, and then our value, and then comma, and then our next key, and the value, and a comma, and so on. So if we have, if we press play, what's happening here is we're creating this dictionary with our location keys and our Major League Baseball team values. And if we want to print, if we want to see what the keys are, we do, we print the name of the dictionary .keys with open brackets. Sorry, not open brackets, with the brackets next to it. And then if we want to get the values, which are these, we print the dictionary name .values. And then if we want to see what type this is, then again, we can use the type function to get the type out. So once we do that, the keys are, as we expect, the locations here, the values are the teams, and the class is a dictionary. So you can also try creating your own dictionary. So maybe you can try adding a new value. I mean, to be honest, I don't know the Major League Baseball teams off by heart. But if we can then put in, so if we want to add a new value to the dictionary, we need a comma, and then we can just add a new line. And you can see here that now, once we print the keys and the values, we've got a new key, which is the one that we just added, as well as a new value we just added. So I've gone through now the kind of first little subsection. So again, I'm going to ask if anyone has any questions so far to please put that in the chat, and then we'll be able to kind of get to that. So I'll just go back and just refresh exactly what we looked through. So we started with some basic operations. We talked about how putting a hashtag means that the code isn't run. We talked a bit about variables and how they're just used to kind of store information, and that we can perform functions on variables. 1 second, I think I'm getting a question. Okay, no, I'm getting it, that it's all good, that's great. So yeah, we also showed that we can print things as well. We showed how you can import a package and how you can import it as its alias, so we don't have to type numpy every time. We showed how to create lists and arrays and how to get their types, how to get the length, and how to extract certain elements from the list. We also learned how to define a function and use for loops to iterate through arrays or lists. We demonstrated how the function works. We also created a dictionary, and also again showed that the dictionary's got keys and values, and if you print the type, then it's a class called a dict. So now I'm going to move on to pandas. So pandas is another package. So again, same as NumPy, pandas is a Python package, and we can also import it, and we're using its alias PD. So pandas is a really, very useful package because it makes the manipulation of tabular data very easy and very intuitive. So if we have data in lists where each list contains data for one column, what we can do is create a data frame from scratch. So how we do this is we create a dictionary, which is what we just went over here, which maps the column names, which are the keys, to their corresponding lists or values. So if we recall the format of a dictionary, we have the curly brackets, and then we have a key 1, value 1, comma, key 2, value 2, and so on until the last key and the last value. So let's put this into practice. So let's make 3 lists. We want to have a data frame with 3 columns. List 1 here, I've said I put ABC. List 2, I put 1, 2, 3, and list 3, I put X, Y, Z. So then we need to define the dictionary, which maps the column name to the values. So here, these are our columns, and we want to put those into the dictionary where the key is the name of the column. So we're making a dictionary here called coldict. So again, as you can remember, we have these curly brackets, which denotes that it's a dictionary. We have the format of our key, colon, value, column, key, colon, value, comma, key, colon, value. And then to create a data frame from a dictionary, we can call a function called pd.dataframe. So we're using the data frame function in the pandas package. And as the input to the function, we're putting in the dictionary that we just defined. So if we press play here, you can see that we've created a data frame where we have our columns here on the top. These are the rows, and they have indexes of 0, 1, and 2, because if you recall, in Python, we start from 0. And you can see that each of these columns now contains the contents of their corresponding lists. So we can also actually print, let's print coldict as well. Oops. So you can see now that that structure of a dictionary, that makes sense, because we've defined the variables, we've defined the lists here. So although here we're writing column 1, list 1, what it actually is taking the values that we've defined for each of the lists. So then when we create the data frame, it reads column 1, and then list 1, it knows that it's these values here. And because, so if we, for example, removed the colons from here, we would get an error because we have nothing called column 1. So if we want to just put in the string, then we need to surround it with the inverted commas here. But if we're just declaring a variable that's already defined, we don't need to put any colons around it. We just put the name as we've defined it here. So that has fixed that, and we have our data frame. So to load data, so it's very common that we'll already have a data set that we want to load in and have it in this format. So the way that we do that is using a function called readCSV. So there's some data that's already kind of initialized in the workspace it's in a folder called sample data, and it's a file called California Housing Train.csv. So we can use the pandas. So again, we imported pandas as its alias PD. So we access the function using pd.readCSV. And what that does is it reads in the file as a data frame. And we can use this other function called info to get some information about the data frame. So if we run this function, the data is read in, and we get some information here. So the information that we get is the number of entries, the number of columns. So yeah, the number of rows is 17,000 and the number of columns is 9. And so here we have it says non-null count. So here it's saying that we have 17,000 non-missing values for each of these columns. So this is very useful when you're modeling data, because you want to see if you have if you have missing data. Here there is no missing data, which is great, but there'll be situations where there'll be things missing, which we'll demonstrate when we do the actual modeling. So yeah, and I've said this here. So there's functions to easily remove missing values, which we'll go over soon. So what this data set is used for is for modeling this median house value. So for this case, these are the descriptors, these are the features of the model, and the median house value is the target. So what we want to do is we want to kind of extract just the target itself. So when we're performing modeling and we want to tell the machine learning algorithm, here are our descriptors, here is our target, we want to be able to separate the two out. So how can we do that? So first, let's see what columns we have in our data frame. So to do that, we put the data frame name, which we've declared as df.columns. And if we play that, we get all of the column names that are in that data frame. So we want to create 2 new data frames. One for the features, which are here, so columns 0 to 7, and one which is just got the house value. So how do we do that? We want to subset the original data set, so the original data frame, which we've already declared as df. To subset the data frame, we need to use these outer brackets, and we want to use a list of column names to subset the data frame. So here, if we say we just want the target or the y variable, we put the data frame, which is declared as df, and then square brackets, and then a list of the values that we want. And in this case, it's just a list of 1, which is the median house value. And then on the other hand, we want the other columns. So again, we define a new data frame called df descriptors, where we're subsetting this original data frame, using the square bracket to subset, and then using a list of column names to define what we want to subset. So now we're just putting in all the other column names. And then if we run this, we can print the shape of a data frame. So the shape will tell us how many rows and columns there are. So both data frames have 17,000 rows, which is what we expect. We weren't subsetting on the rows, but the number of columns, which is a number here, is different. So because we've just taken the target value here, median house value, we have 1 column, whereas for the df descriptors, we've taken these 8 features or descriptors, and we have 8 columns here. So we've read in our data frame, we've seen how much data we have, how many missing values we have, how many columns we have. we've printed what the columns are, and then we've subset the data frames using the different column names that we want. So because this is a 1D data frame, so we know it's 1D because it just has a one column, we can convert this to a NumPy array. So to convert a 1D column, so one column into a NumPy array, what we can do is we can take the data frame and then say dot values, and what that will do is extract all the values that are in that column. So we can print, I can show you at df target. Looks like this, and then the values from that are is this array. Now, as you can see here, it's what's called a nested array. As you can see that there are kind of brackets around here, and what we want is kind of a flat 1D array. So if we go back to the array that we made before, you can see that it was flat, so it's just one set of square brackets, so 1, 2, 3, 4. We want to be able to convert our data to the same thing. So to do that, we're having on. To do that, we use a function called ravel. So this is again from NumPy. So we want to take df target values, which is this nested array, and we want to flatten it. So we call NumPy or MP, because that's the alias that we imported, dot ravel, which is the function, and we're using as input this array here, which is the one-dimensional one-column data frame and its values. So once we run ravel on this, we get this kind of flat structure, which is what we want. So you'll notice that we have kind of created new variables for our subsets. This doesn't affect this initial data frame that we read in the first place. So if we print df.head, so df.head is taking the first 5 rows of our data frame, you can see that it's remained the same because we've created new variables for the 2 subsets. So is everyone following? Is that okay for everyone so far? So read in the data frame, got the columns, subsetted them for the features or the descriptors, and also for the target. We converted this into a 1D array, and then we've also shown that our original data frame that we read in hasn't changed because we've declared new variables for the subsets. So I'll just check that everyone's finding that okay, and I will check and see if I have any questions here. So yes, Srijit will be replying to you if there's anything. Okay, so I will continue. So what we can also do with data frames is create summary statistics. So we can take a particular column out and we can take the mean or the median. So here we're printing the longitude columns mean and the latitude columns median by doing df and then putting in square brackets the column that we want. And then the function dot mean with the brackets means that we're taking the mean, and then the same with the median. So if we play that, we get the mean and the median for the 2 different columns. But if you want to get a summary of all the columns, there's a really nice function called describe. So again, we're using the original data frame and we're using the dot describe function. And if we play that, we get a summary of the count, mean, standard deviation, min, and then different quartiles for each numerical column that we have. So this is a really good way of kind of looking into distributions of your data using pandas. So that's it on pandas. I'm going to be moving on now to RDKit, scikit-learn, and Seaborn. This is the kind of last bit before I start the actual hands-on modeling. The reason that I'm taking you through these packages is because they're kind of specialist packages. RDKit is used for cheminformatics analysis. So it's used to generate descriptors from molecular representations, as Andreas was saying. Scikit-learn, often called by its alias sklearn, to actually carry out the machine learning. And then Seaborn, which we use its alias SNS for plotting, for making plots and visualizations. So now I'll just briefly introduce each of the packages. So RDKit is a cheminformatics package used in Python. So most of the functionality of the RDKit package is in a module called chem. So in Python, distinct collections of functionality are kept separate as modules, which contain submodules or functions. So we want to kind of import these submodules to be able to gain access to the functions that are within them. So to do that, we need to, from RDKit, we're importing the chem module, and from the chem module, we're importing the allchem and draw modules, which is what we're going to be using now. So if you run that, it imports everything that we need, and now we can access all the functions that are within them. So RDKit can be used to read in chemical information in several different formats. So for example, SMILES. So SMILES, if you're not familiar, stands for Simplified Molecular Input Line Entry System. And it's just one of the few ways that computers can read chemical structures. I've provided a web link in the Colab notebook, which explains what SMILES are in a bit more detail. So here what I'm doing is I want to read in a set of SMILES, and I want RDKit to be able to kind of do certain functions to it. So to do that, the SMILES have to be converted from a string, which is what they are here, just a string of letters and numbers, into a kind of internal representation that the package can use and do stuff with. And in RDKit, this is called a mol object. So what a mol object is just an object which is held like internally, and we can't really see it here. So if we read in, so we're using the function from chem called mol from SMILES to generate the mol. So chem is the module that we've loaded, and mol from SMILES is the function, and then the function takes in a SMILES string. And if we declare that as a variable mol, and we say what mol is, you can see that it's RDKit mol, and that kind of that's all it really tells us. But we can use that mol object and manipulate it and do certain things to it. So for example, we can draw the structure directly in the notebook. So to do this, we use a function from this draw submodule, which we imported, called mol to image. And what mol to image does is it takes the mol object, which we've declared here, which we've read in from these SMILES. And if we press play, it draws the chemical structure as an image. So mol objects can be converted back to SMILES, which is quite useful. So you input your SMILES as a mol object. RDKit, you use RDKit to do various things to it, and the various things I will mention in a second. But then you want to convert that back to SMILES at the end. Well, the way to do that is a function called mol from SMILES. So as you can see, we have mol from SMILES to read in from, get the mol from the SMILES. So mol from mol to SMILES then takes the mol and turns it to SMILES. So what we're doing here in each of these lines is we're saying we want to print the result of the function mol to SMILES applied to the mol object, which is generated from the mol from SMILES function. So mol from SMILES generates a mol object, mol to SMILES converts that back to SMILES. And it might seem a bit counterintuitive why I'm doing this here, but what I'm trying to show you is that RDKit will by default output what are called canonical SMILES. So what this means is that the same structure will always have identical SMILES as their output by RDKit. So if we put in, so here I want to read in 3 different SMILES strings that are actually representing the exact same molecule. And if we read it in with mol from SMILES and then convert it back to SMILES with mol to SMILES, you can see that the output for each instance is exactly the same SMILES string. So just to recap what we've done here quickly with RDKit, we've imported the modules that we want, which is chem, allchem, and draw. We've read in some SMILES and converted them to a mol object using the mol from SMILES function. We've shown the mol to image function, which works on this mol object and draws the molecule there. And we've also converted the mols back to SMILES and shown that by default, RDKit will output canonical SMILES so that the same structure will always have the same SMILES. So what can RDKit be used for? So RDKit can be used for many things, and one of them is calculating or predicting various molecular properties. So we're going to be using RDKit descriptors in the first part of the workshop. So these are the physical chemical descriptors that Andreas was discussing. So I'm going to show you an example of how you can calculate descriptors, a small subset of descriptors. So we need to import a submodule called descriptors, which is present in the chem module. So from rdkit.chem, we're importing descriptors. We're creating our mol object, again, using mol from smiles, where we've put in the smile string here. And then what we're doing is we're using the descriptors module to calculate the mol log P, TPSA, and exact molecular weight for the molecule. So if you print that, you can see that each one of these corresponds to each of these descriptors for that compound. So in the workshop, we're going to generate a larger array of descriptors to use as input for machine learning. So that was RDKit. Let me know if, well, let the chat know if you have any questions about that. Essentially, we just went through the fact that it's a way to kind of read in chemical structures and manipulate it in certain ways. So for example, printing them as an image or generating descriptors from it using different functionalities that we have. And they all involve converting the structure into this mol object, which is the kind of internal representation that RDKit works with to apply its functions. So what I'm going to go through next is scikit-learn and Seaborn. So it's the last thing I'm going to go through before I take you through the modeling. So scikit-learn is another Python package, and you can see how great Python is and how it's really great that there are so many open source packages to be able to do all these very, very complicated things. So scikit-learn is a package which is used to carry out machine learning. So it's imported usually using its alias sklearn. Oh no, it is imported in all cases using its alias sklearn. So earlier we created two variables. If you remember, we created array, target, and df descriptors. So if we go back and see what they were, we've created two variables, one which is the target, so what we want to predict, which in this case was the housing prices. And we also created a data frame which has the descriptors, so the original data frame without the target variable in it. So how do we use this to actually create a model? So the first thing that we're going to do is kind of split the data into two sets. So as Andreas was saying, we need to separate our data into training and tests. There are various ways of doing it, such as cross-validation, but here we're going to do the most simple way, which is just one split where we take a certain percentage of the data set and we say this is our training data set, which is what the model is going to be fit on. And then the rest of the data is kept as a test set, which is what we then evaluate the model on and how we measure its performance. So to split a data set into train and test, scikit-learn has a function called, very handily called train test split. So this is present in the model selection module of scikit-learn. So from scikit-learn model selection, we're importing train test split. So what we need to do is, as an input to train test split, we want to put in our X and our Y. So our X is our descriptors and our Y is our target or our label. So here that's df descriptors is X and array target is Y. We also need to say how big we want the test set to be. So here we're saying 20%, which means that will be 80% will be split into training and 20% will be split into test. And what we've also done here is put a random seed, which means that it will always be the same split if you run this function on this particular data set with that particular seed. So what the outcome of this is so obviously we've put in like two things in here and we're getting out 4. So we've put in an X and a Y, and what we're getting out is an X train, X test, Y train, and Y test. So X train is used to, X train and Y train are used to fit the model. X test is then, the model is then applied on X test and it's compared with Y test. So that's what all the different data sets are used for. So if we run this, we can see that, so now we're printing, so for I. So if you remember for loop, what it does is it goes through every element of a list. And we're saying for each element in this list that we've declared here, which are the X train, X test, Y train, and Y test that we just created with train test split, we want to print the shape, which, as you remember, is the number of rows and number of columns. So as you can see, we have, so it was 17,000 data points. We've now split into our X train and X test. We have 13,600 training points and 3,400 testing points. Our X's have got 8 columns because those are the descriptors, and the Y's, which are our label or our target, have no dimension because they're flat arrays, as you remember, as we created with the NumPy rabble. So now we have our 4 new variables, which is X train and Y train, which are used for training, X test, which was used for applying the train model, and Y test, which is used for evaluating the prediction using the train test split function. We want to now fit a model, and with scikit-learn, it's really great because it only takes a few lines of code. So I'll take you through what that code is. We want to perform a random forest regression because let me print our Y train, for example. So you can see here that this is money, this is house prices. So this is a regression problem. So we're going to be using a random forest regressor. So to get access to that random forest regressor, we need to import it from the submodule called Ensemble. And the reason that the submodule is called Ensemble is because, as Andreas showed you, random forests are ensembles of decision trees. So we're from scikit-learn Ensemble, we're importing the random forest regressor. We want to evaluate how well our model is performing. So we need to define a metric. So as Andreas said, there's lots of ways to do this. In this case, we're just using the R squared. So from scikit-learn metrics, we're importing the R squared score, sorry. So how do we use these functions to actually train our model? So the first thing we need to do is define what regression model we want. So it's the random forest regressor. So we define a variable called reg for regression. We're saying that this is the random forest regressor, which is what we're using. To actually train the model, we just take this reg that we just defined and we use .fit. And recall that when we're fitting the model, we're using the training set. So we want to fit X train on Y train. So we want to fit the descriptors on the variables. Once we've fit the model, we want to then apply it. So as you can see, we've started with our regressor. At this point here, we've now trained our model. So now we can use this model reg to predict on X test. And then we call that Y pred. And then we can use the R squared score function to compare the test set, so the true variable, the true values with the predicted values. So I'll click run and I'll just go through that again. So we have our train and test split. So we created our X train, X test, Y train, and Y test. And we're using them at the X train and Y train to fit the random forest regressor. And we're using the X test, and we're using the model to predict Y pred using the descriptors that are in X test, and then we're using R squared score to compare the true values, which was Y test, with the predicted values, which is Y pred. And you can see that the output here is we have an R squared score of 0.82, which is very nice. So can I go through? No, I think I'll keep going for now. And then I'll do another recap after this. So if we want to visualize the predicted versus actual results, so as Andreas was saying, correlation coefficients are misleading. We need to look at the distribution of our data and what it actually looks like. So Seaborn is another great package, which is good for visualization. So we're importing the Seaborn package with its alias SNS. And when we do that, again, we gain access to the functions that are within it. So we want to create the plot. What we need to do is we need to create a data frame which has the predicted and true values. So we want one column with the true values, so our actual values that have come from the initial data frame. And then we want another column, which is the values that we predicted using our model. So how do we do this? Well, we know that we can create a data frame from a dictionary where we provide our So we define a dictionary with the curly brackets, and we define our keys and colon, and then our values, where our values are here in this case a list of the true targets, the true labels, and predicted has its value is the list of predicted values. So we can wrap that around the pandas data frame as we did before. And if you recall, putting dot head just prints the first 5 rows. And actually, if you want to make this first 6 rows, you can put 6 in there, and you'll get more rows out. So we've used this now, and we've created a data frame, 1 column for our true. So this is the actual house prices, which will come from the data frame, the original data frame, and predicted is what we've predicted using our model. So now we can pass this data frame to the Seaborn function. So we're using the scatterplot function from SNS of Seaborn, which we imported as alias SNS to create a scatterplot. So to create the plot, we need to first of all put in the data frame, which we've done here. We've defined it here as pred_df. So we put that in, we say data equals pred_df. And then we want to just say what the X and the Y is. So the X on the plot is the true values, and the Y are the predicted values. And when we press play, you can see that it plots a scatter plot of our predicted versus true values. So I'll go back and just quickly recap what we did here with scikit-learn and Seaborn. So scikit-learn is a package for machine learning. It has a function called train test split, which can be used to split our data into training and test sets. When we perform the train test split, we get 4 values that come out of it, which are the descriptors for our train and test set and the labels for our train and test set. And you can see that it split them such that we have 80% in the training set and 20% in the test set. We've used random forest regressor, which we've imported from the ensemble model to first of all declare a random forest regressor and then use it to fit it using the training data. We then applied it on the testing data to obtain our predictions, and then we used the R-squared score function to obtain the R-squared score by comparing the true values with our predicted values. And then finally with Seaborn, we again created a data frame from a dictionary using our predicted and true values. And we've passed that to the Seaborn scatterplot function so that we can visualize our true versus our predicted values on a plot. So this was just a sort of introduction. I'm going to get into the actual modeling of the tox data now. So you should be familiar now after this section with basic concepts of Python and also Google Colab notebooks, including operations, variables, lists, arrays, dictionaries, and functions. We also explored using pandas data frame for manipulating tabular data and briefly introduced some functionalities of the RDKit, scikit-learn, and Seaborn packages. So hopefully everyone's going okay with that. I haven't had any messages from anyone saying that I need to kind of say anything here. So what I'm going to do is I'm going to move on now to the modeling exercise, and then of course at the end there'll be time for questions that we can kind of participate in or discuss them together. So exercise 1, what we're going to be doing is using random forest to model hERG inhibition in the form of IC50 data. So if you can recall, the very first thing that we did at the beginning of the workshop was we downloaded some data. So me and Srijit have already prepared the data sets, which are in this GitHub repository, which has been kind of downloaded to your local drive here for this Colab notebook. So if I go down back to the modeling exercise, the file name that it's been saved as is hERG_IC50.csv. And what this file contains is IC50 data for hERG, which I extracted from the Campbell database, which is free and very big public database with all sorts of bioactivities and different organisms and so on. So here I've extracted the hERG data. So if I want to read in this file here, I'm using the pandas read CSV function, and I want to print the first 5 rows. So When we do that, we've read in the IC50 data and we've printed the first 5 rows here. You can see that there's a lot of information in this data set. A lot of it we don't really need. So to make our lives a bit easier, let's just take the columns that we actually care about, which in this case is going to be the SMILES, which is the compound representation, the standard relation, which is, whether it's equal or greater than or less than, and then the p-Chembl value, which is the negative log 10 of the molar IC50. And the reason that we want to model the p-Chembl value is because putting it on a log scale is easier to model than the raw IC50 values themselves. So what we want to do is subset the data frame to only contain these columns. And we know from the pandas exercise we did that we can subset a data frame using the square bracket. And then inside the square bracket, we have our list of columns that we want to keep, which again is SMILES, standard relation, and p-Chembl value. And if we subset it, we're defining it here as a new data frame called HERG data dropped, because we've dropped some columns. And then if we print the first 5 rows, you can see that we've lost these columns. And then again, if we want to print the original data frame, which is HERG data, this has stayed exactly the same. So that's how the variables work. So we can quickly summarize this data frame using info. So if we run this, this is where the modeler would start to go, oh no, we have missing data, we need to do something. So if you can recall, we have this non-null count, which shows how many times we have a value which isn't null. We have 14,364 entries in our data frame in total and 3 columns. And you can see that we have missing values all across for all of the columns. So we have 14,364 entries, of which 14,332 have SMILES, 12,542 have a standard relation, and 8,744 have a p-Chembl value. So we can't use all of these entries for machine learning because we can't do anything with the missing data. So we need a data frame which only contains rows where every column is non-null. So we need the SMILES to be able to generate the descriptors. We need the p-Chembl value because that's the label. And we need the standard relation because then that tells us what the data is, essentially, what the value is, if the value is kind of greater than or equal to or less than the p-Chembl value that's stated. So if we want a data frame that only contains rows where all of these columns have non-null values, there's a function in pandas called drop NA. So how drop NA works is it takes a data frame and it will drop either non-null rows or columns by default rows. And there's 2 arguments you can set for this function called it's called how. You can have any or all. So if how is set to any, it means that rows which have missing values in any column are dropped, which is what we want. The other option is all, which only removes rows if all of the column values are empty. So if we had a data frame and it was for SMILES, standard relation, and p-Chembl value, there was nothing there, all would remove that. But then if we had, say, only one row missing, all would not remove those rows. We need any because any will remove columns where any value, remove rows where any column value is null. So if we run, if we use the HERG data dropped, which is the columns that we subsetted out, and we use the drop NA function with how set to any, what happens is that it goes through each of the columns, it goes through each of the rows and it drops ones that have null values in any of the SMILES, standard relation, or p-Chembl. So once we run that, we can see what's happened. We've ended up with 8,744 rows. So that means that we've lost quite a lot of data. So if we want to just show data drop NA. So in this data frame, actually we can try this with .info. So if we use .info, which, as you can recall, gives us the information on the data set, we can see that for each one of these columns now in our new data frame called HERG data drop NA, we have 0 non-null at all. So we have an entry for every single one of these columns in this data frame. So the next thing we want to do is we only want to include data for modeling if the standard relation is equal, because the way that these assays work, sometimes you'll only have that it's greater than a certain value, but we don't know if that is the exact value, so we can't really use that for regression. So we only want to keep measurements where we have an equals in the standard relation. So we can check which standard relation symbols are in our data frame by using a function called unique. So what unique does is you can take for example, a column, which here is our hERG data where the NA is dropped, and the standard relation column. And if we call .unique, we get the unique values that are in that column. So if we print that, we'll see that we only have equals. So that's good. So we don't have to do any more cleaning there. But if we did have the greater than or the less than signs, we would have to remove those rows. So the next thing that we need to do to clean our data is deal with duplicate values. So it's likely that in Chembl, there's many assays run on the same compound. So one compound could have multiple values for a particular endpoint. So the way that we can check how many duplicate values we have is using a function in pandas called group by. So with group by, we can group the data frame by a certain column and then perform different functions on it. So we're calling the hERG data drop NA. So this is the data frame with all the null values dropped. We're calling group by, and we're grouping by the smiles column. And then we're saying what size, so how many do we have? And then we're saying sort values in descending order. So when we run this, you can see that here we have this output here it's not a data frame, but it looks a bit like a data frame in that we have our two columns, which is smiles, and these are kind of unique smiles throughout the data frame. And what this says is the number of times that it appears. So grouped by the smiles, we've got the size, which is the number of entries, and then we're sorting values in descending order. So you can see that this compound here is present in the data set 24 times. And the length of this is 7,412. So out of the 8,744 rows that we only actually have 7,412 unique smiles. Now we want to deal with these duplicates, and there are several ways of doing this, but for this exercise, what we're going to do is we're going to say that if we have a compound and it has multiple measurements, we just take the mean. So to do that, we can again use group by. So if you recall here, when we group by smiles, the output that we get, only the smiles column is kept. So here we want to group by smiles and standard relation, and then that means that the column is kept. So we are using the group by function, grouping on the columns smiles and standard relation. And then we want to take the p-Chembl value and then return the mean. And we're calling that, we're defining that as the variable hERG data cleaned. And then we turn it back into a data frame format. So as you can see that this isn't exactly a data frame, but can be converted very easily into one using the PD data frame function. And then we're printing the first 5 rows. So if we print this, it's grouping the data frame by the smiles and standard relation. And then what happens is we have then, so say for this smile here where we have 24 entries, what this has done at this point is we have the smiles in each of the 24 entries, and then we're taking the p-Chembl value column and we're taking the mean, and then that's now being put back into the output. So then if we turn it into a data frame, you can see that we have our smiles, our standard relation, and our p-Chembl value. And if we print the shape, you can see that we have 7,412, which is what we want because those are the number of unique smiles that we have. So because we use the smile and standard relation columns for the grouping, because we wanted to keep the columns, these have now become indexes in the data frame. And the way that you can tell is you can see here the shape here says we only have one column. Okay, cleaned. So it shows that we only have one column, which is p-Chembl value, because of the way that the grouping works. So we want to turn these back into kind of regular columns rather than indexes. So to do that, we use a function called reset index, which just changes them back into normal columns. So if we run reset index and we print the shape, you can see that we have 3 columns again, and we can actually print the data frame itself. And you can see that we've gone from having indexes, smiles and standard relation as indexes into kind of regular columns. So now we have our non-duplicated data set of 7,412 unique compounds. So I took mean in this case. There are other ways to deal with duplicates, for example, discarding outliers or taking the median. So let's see what the distribution of p-chem values looks like. We can use the violin plot function in Seaborn to do this. So what we need to do is we have our data frame, which is Herg data cleaned, and we want to plot the distribution of the p-chem value. So we put in our function sns.violin plot. We say that our x is Herg data cleaned p-chem value column. And then when we press play, a violin plot is created of the p-chem values. So from this plot, we can see most of the values fall around 5, and they span from about 2 to 10. Again, this is the kind of exploratory analysis you should be doing with your data before you start modeling. And we can also use that describe function that I went over earlier to summarize the p-chem value distribution. Now, if you recall, describe only works on numerical columns. So now it's outputting the count and the mean, which, as you can see, corresponding from the plot, is about 5.43. So we have a clean data set now. I'm going to go back and just summarize what we did here with the cleaning. So we loaded in the data as with pandas read CSV. We kept only the columns that we need for modeling. We used info to summarize the data frame, and we found that we had missing values. So we used the drop-NA function with how set to any to remove any rows that have any missing values in any of the columns, left with 8,744 rows, which have no missing values at all. We checked and we saw that our standard relation is equal in all cases, so there's nothing we need to do there. And then we also saw that we had duplicates in our data set by using group by. And we dealt with those duplicates by taking the mean of the p-chem value, again using group by, and then taking the mean. And then finally, we turned this group data frame. We kind of rejigged this group data frame from having the columns as indexes back into actual columns. And then we plotted the distributions of the p-chem values as well. So now that the data set is clean, we can generate our features. So we're using the RDKit descriptors to featurize the compounds. So this code will assign a list of available descriptors to a variable called descriptor names, descriptor list, that's incorrect, and prints the length of the list. So from RDKit chem, we're importing descriptors. We're taking every descriptor name that's in the descriptors in RDKit, and we've got a list now of 208 descriptors, which you can see here. If we show it here, are various kind of physicochemical descriptors that are in RDKit. So now we need to set up a calculator to calculate the descriptors. So the descriptors module has a molecular descriptor calculator. So what we need to do is we've got our list of descriptors, which we already declared here. We set up a calculator using calc, which we've said is the molecular descriptor calculator, and we're calculating the descriptors we define in the list. Once we've set that up, we can calculate all the descriptors for a given smile string by doing the following. So we need to read in the smiles as a mol object. We then calculate the descriptors from the mol object. And then if we print this, you can see that we end up with the descriptors. So by running the calc descriptors, which we set up with the list of all descriptors in RDKit, we're able to obtain all of the descriptors for a particular mol object. And you can see that these are the descriptors and that it has a length of 208. So now what we want to do is we want to create features for all of our smiles. So to do this, we're creating a for loop. So we've taken all the smiles from our clean data frame, and we've used .toList to put them into a list. We've created an empty list, which is what the descriptors are going to go into as they generate. And then again, using a for loop, we are iterating. So for each element in the list, which is for each smile in the list of smiles, we're generating the mol object, we're calculating the descriptors, and then finally we're appending the descriptors to this empty list, which will become populated as the loop executes. So this takes a little bit of a moment to run. So I'm going to keep going while that's running. So what we want is descriptors in the format of a data frame. So what we have here is a list of lists, because what comes out of the descriptor calculator is a list of descriptors. So to create a data frame from a list of lists, we can again just call the PD data frame function. And what it will do is it will create a data frame where there is a column for each one of the descriptors, and there's a row for each column. Now you'll notice that the data frame that we created hasn't got any column names. So we'd quite like to add those back so that we can kind of interpret the model at the end. So to do that, we can define the columns of a data frame. So our descriptor data frame dot columns, we can set as equal to our list of descriptors, which were the descriptor names that we extracted here. So we just set the column names of this data frame to the descriptors. And now we can see that we have rows for each compound. We've taken heads, so only 5 rows are showing here. And the columns are the descriptors. So now we have everything in the correct format for modeling. I'm going to go back a bit actually, because this is still taking a bit of time to run. So I'm just going to go over what we've done. So we've imported our descriptor model. We've extracted a list of all the descriptors we can calculate from the descriptors module, which are these here. And there's a few of them. We've set up the calculator to calculate the descriptors, and then we've used using the molecular descriptors module, molecular descriptor calculator function. So now this is saying we've kind of set up a thing and we say we want to generate descriptors for all the descriptors in this list. And then we've applied this calculator to, in this case, an individual mol object generated from smiles. And then here we've generated them for all of the smiles in our data set using a for loop. So we've gone over each of the smiles and we've generated the descriptors. So this is run now. So now I can run these and show you that we've created data frames from the descriptors, or a data frame for the descriptors, and we've appended the descriptor names as column names. So now we have our features in the correct format for modeling. When we calculate descriptors, it's possible that some calculations will fail, which means that we'll have null values for some compound descriptor combinations. So to remove columns where there are null values, we can use drop in A again. But this time we want to remove columns, not rows. So we need to pass a new variable called axis. A new argument called axis. So if we set axis equals 1, it tells the function that we want to drop columns, not rows. And then we're using any again, because we want to remove columns where there is any missing value in any of the rows. So when we run that, we can see that we dropped 8 descriptors because they had missing values for 1 or more of the rows. So now we've cleaned the data and calculated our descriptors, we can proceed with the modeling. So we start with performing a train test split. So we need to get our labels. So we get that from the data frame, hug data cleaned. So we extract. So if you can recall, we use, we take the data frame name, the column that we want, dot values, and that forms a nested array. We use NumPy Ravel to turn that into a flat array, which you can see here is now an array of the pChemol values. Now we can use the train test split function to define our X train, Y train, X test, and Y test using again an 80-20 split. So we put in our descriptors, which is our descriptor data frame that we've created here. We put in our pChemol array, you've said what our test size is, and then again, we've put a random state, which means that when you run it, you'll get the exact same results. So now that we've split the data, we can train a random forest regressor, which is, again, just a few lines of code. So we just say, we're importing the random forest regressor. We're saying our regression model is a random forest regressor, and now we're fitting it on the training data. So fitting the descriptors on the labels. And then once that fits, which again takes a moment to run, we apply the model to the test dataset descriptors, which from our train test split function is x test. And we want to store the results in a variable called ypred. So if you can recall, once we've trained a model using .fit, we apply it to a data set using .predict. And then we can say that our predicted values are the predictions that we generated on our test data set. And then we run that, we get, and it's run. When we run that, we get a set of predicted values. So these are the predictions of the p-Campbell values from the model that we've just built. So how well has it performed? So earlier I showed you the R-square score metric. There's also the mean absolute error. So here I'm printing the R-square score and the mean absolute error for our model using the actual label y-test and then ypred, which is what we predicted. And once we print it, we can see the model has an R-square score of 0.51 and a mean absolute error of 0.44. And we can visualize again the true versus predicted values by creating a dictionary, creating a data frame for a dictionary and using the scatter plot function. And then this is the plot that we have. So model performance is not great, but we can improve it using hyperparameter optimization, which is going to be covered by Srijit in the next exercise. So we can interpret models in a few different ways. So here we're going to talk, we're going to show how to do it with SHAP values. So SHAP values is a score that assigns the importance of features in the model. So the decision trees are very good for interpretable and explainable machine learning because you can kind of follow the tree structure and what's happening at each decision point, which is what Srijit's going to show. So the SHAP value shows what contribution of each feature to the model. So to understand the contributions of each feature to the model, we're using a library called SHAP. So we're importing SHAP. I'm not running this because it does take a while to run. It takes about 10-15 minutes, so I'm not going to run it, but you can run it and then you can see the output here. So to explain the predictions from a model, so we fit our model in this code here. We fit it to the training test set. So we create an explainer. We use the tree explainer function from the SHAP module on the regressor. And then what happens is it fits the explainer using the train model, and then we compute the SHAP values or the importance values on our test set. And then once we plot that using the summary plot, we end up with a plot that looks like this. So the way that this plot is interpreted is that red are when the values are high and blue are when the values are low. And then this line shows the impact on the model output. So whether it makes it a negative prediction or a positive prediction. So you can see, and it's also ordered by importance. So the top two most important features here are the TPSA and the log P. So how you can interpret it is that as the value of TPSA increases, the predicted hug PIC50 decreases, and vice versa for more log P. So you could say that, according to our model that we've interpreted, if we want to develop a new compound without hug, we should ensure that the TPSA is high and the log P is low. Of course, in reality, it's not as cut and dry as this. It's not as simple as doing this, but this is a good starting point to try and understand, why we're getting the predictions that we're getting, which can then maybe in the future inform us to try and design molecules that don't have this liability later on. So in this workshop, you've successfully trained a hug prediction model and you've visualized the feature importances. And I'm going to pass over to Srijit now, but if you have any questions, please keep them coming in the chat. And while Srijit is talking, I'll try and answer your questions, and then we can have some time at the end as well.
Yeah, I'll just try share my screen quickly. Does this work? You see my screen, Layla?
Yeah, can see it.
Yeah. So the second part of this project, we are trying to build a classification model for mitochondrial toxicity. And in this classification model, what we're trying to do is predict whether a compound would be toxic or non-toxic. So the first, we load all data sets. So RDKit for chemical structure, C-bond for any kind of visualization, along with matplotlib, which is also for visualization purposes. And then you have pandas for data frame related work and NumPy for the array related calculations. With wget, we get a data set which is stored in my GitHub repository. And then with the df pdread CSV, we load the data set, as you can see. So in this case, you don't have smiles anymore. We have another kind of representation known as INCHI code. The INCHI code here is already standardized. So, and the activity readouts for around 6,500 compounds here. So first we load the activity count values, and we see there are 5,544 compounds which are not active and 916 compounds which are active, which means our data set is quite unbalanced. It's 1:6 or something like that, right? So below there are two functions to first develop smiles from the INCHI code. So as you can see in this case, we are trying to one second. Yeah. So we are trying to develop the smiles from the INCHI code. So here the first function, we have defined two functions here. The first functions returns a mole molecule, a smiles from an INCHI molecule. So it gets a value, which is an INCHI code, derives a mole from the INCHI, and from the mole, it gives you a smiles again. And the second one standardizes these smiles. So once you get the smiles code, these smiles might be, some of the compounds might have similar structures, but they have different INCHI codes. So we need to standardize the smiles. So we do things like disconnecting metal atoms, normalizing the molecule, ionizing them, removing if there are two fragments or if it's a mixture, we only take the actual molecule we're interested in, the largest fragment, for example, here. We try some automerization, etc. So and at the end you return a particular smile. So you can use this and you can follow Greg Landrum's tutorial on how this actually works. So this next command actually then takes the INCHI code and first builds you smiles. And in the next command here, the second line takes these smiles from here and returns you some standardized smiles code. So at the end, you get a set of smiles, and you see from here to here, you get you remove the protons excess and whatever, and get a standardized smile set. So I'm not going to run these again, but we remove the compounds which we could not standardize smiles. So you can see there are some returning cannot do for some reason. If it is corrupt or so, you do not get any readouts. You remove the compounds which failed here. And then we sort according to activity. So the highest active compounds are on the top, and low active compounds, which are non-toxic compounds here, are at the bottom. As this is the classification, you only have two classes here. And then we try to say there are some duplicate activities. Now that you have removed some functional groups, or you've removed salts, for example, and you have protonated them at a particular pH, for example, then there might be some duplicates. In that case, what we have done here is we have chosen evidence of activity or activity for a compound. So if there are two duplicate records, one which is active, and say this record is the same as the one here, but this one is inactive, we have chosen that the compound is going to remain active. So we have dropped duplicates, and among all duplicates, we have kept the one that appears above in the database. And that means this should be a toxic compound in that case. So after doing this, we are left with 6,432 compounds. So around 12 or 13 of these compounds have been removed. And now we see that we have 5,517 non-toxic compounds. 5,570 non-toxic compounds and 915 toxic compounds. In the next step, we'll generate Morgan fingerprints. And these are more structural fingerprints. They are derived from structures, and you can see the module is loaded from RDKit molecular descriptors, get Morgan fingerprint as bit vector. And what this does is it gives you an array of zeros and ones, which are also binary. And there are 2048 bits of these. So the length of this array on the right here is 2048. So you define Morgan fingerprints using the mole objects. So you have first gathered all the mole object from all the smiles together. So you performed a batch operation from RDK to collect all the mole objects from this data set. And then on all these mole objects, that's 432 compounds, mole objects, you have applied the get Morgan fingerprint. So now you have, for each compound, you have a given Morgan fingerprint of zeros and ones. And what Morgan fingerprints basically are these bits, say in simple terms, correspond to substructures. So 0 could represent a particular substructure, say an alcohol group, for example. 1 could represent, say, the benzene ring. 0 could represent something else. So basically, you get a diversity of different substructures, and this happens in a loop. It's also known as extended connectivity fingerprints. And from here, we therefore get these 2048 fingerprints. And the activities. 1 second, I'll go here. Yeah. So once you have that, we can therefore generate these fingerprint names, which start from MFP0 to MFP 2048, and we'll use these names later on. But we have generated a set of names. If you print this in the next line, you will see it won't run because I haven't loaded the modules here. Then for the next part, you have data equal to df, and the features are the Morgan fingerprint features, and the endpoint is the activity. So I have now defined my test and my features and my endpoint. So features are my Morgan fingerprint in an array like this. So for each compound, I have a set of fingerprints, and y is my endpoint. So for each compound, I have an activity label. It's either 1 or 0. And for each of these, the shape is 6,432 compounds, or 6,342 rows, and 2,048 columns. The next part is where we split the data into the training and the testing set. So in this case, you can see, we'll go back here. 1 second. Where did it go? Yes. So we split the train and a test set. So train and test has been split into 0.25. So 25% of this X data set has now come into the test set, and 75% has gone into the training data. This is what you can see here from the shape. So out of 6,432 compounds, 4,824 are in training, and 1,600 compounds have gone into testing. And the same has happened for the Y labels. So if you print the values of the Y labels, you will see that how many active and inactive are present, or how many toxic and non-toxic compounds are present in your trend and test data sets. The next part which we do is a hyperparameter optimization of a random forest model. So a random forest model classifier in this case can be used to classify mitochondrial toxicity endpoint, and we would perform a 5-fold grid search cross-validation. In this case, what we first do is we first load the 2 modules we need. We need grid search cross-validation and random forest classifier. Then your random forest classifier will run through a grid of different features. So you can optimize the parameters, say 200 to 300 number of trees. The maximum number of features will be determined by either using the square root or log2 of total number of features provided. And similarly, the maximum depth, for example, is how many trees do we look. So we have how many levels of the tree do we look at. So we have chosen between 3 and 5, and you'll see this more clearly in the visualization below. So next we look at the grid search CV, which basically go through this random forest, tries different parameters for 5 different splits of the data. So it splits our training data into 5 parts, so 20% each, and then uses 80% to train on 20%, and then keeps shuffling this 20% around. So you get 5 different values, and out of them, it chooses the best model. So the best model would have a particular combination of these features, which is the parameter that we choose. And then we choose that parameter. In this case, it chose balanced class weight, which makes sense because data was unbalanced. So we need to weight it in some sense to make it balanced. The depth was chosen as 5, maximum log features was chosen as log 2, etc. And then we use these parameters to train on the entire training data. So you have an internal validation. So in the internal training data, we use different splits to find out the best model parameter. And now we use all the information we gained from the training data to train our model on the entire training data. And now we predict it on the external test set. So now the evaluation of the external test set in case of a classification model goes through, say, the confusion matrix. The confusion matrix has the true positive here and true negative here. And this is a false positive and this is a false negative. So what you actually get here is the probabilities from each value. So in this case, unlike a regression model where you get one value from a prediction, here you get 2. The first one predicts, the first one here probability is given as a probability. So that means if the model predicts 0.7, say a threshold of 0.5 is applied, and if it's greater than 0.5, then the molecule is toxic, and lesser than 0.5, the molecule is not toxic. So in the first case, you get a range of probabilities. So you get 0.1, 0.7 for the second molecule, 0.3 for the third molecule. And the predictions would therefore be 0.1 becomes 0, 0.7 becomes toxic, 0.3 becomes non-toxic again. So probabilities are a more finer value, whereas a prediction is a more threshold, applied threshold, and you get a binary or a classification result. So in this case here, when you do a classification report, you can see exactly how many compounds you had for the training and for the non-toxic and toxic classes in your test data set. And therefore the individual F1 scores, which takes into account both precision and recall, and also the balanced accuracy here. So the balanced accuracy in this case was this one here. It's 0.75. So the test, it takes the test set and a prediction. So these are the real values from your real data set, which you haven't shown in the model. And this is what the model has predicted. So you get the balanced accuracy. The Matthews correlation constant takes into account all the 4 classes. So true positive, true negative, false positive, and false negative. And it gives you a correlation coefficient between -1 and +1, where -1 is an inverse prediction. Everything has it predicted the opposite of what it truly is. And 1 is perfect prediction. So everything has it perfectly predicted. And 0 is a random predictor. So you just take a molecule and randomly say whatever you want. That would be 0. And so this statistics also has phi coefficient. And what you get a Matthews correlation coefficient of 0.4, which means it's almost as much better than an average random prediction. Sensitivity and specificity are actually describing the accuracy of a test, depending on the presence or absence of, say, true positives in this case, and then second case, the true negatives rate. So the true positive rate would be how likely is my molecule to predict a true a toxic compound, and specificity would be how likely is my molecule to predict a non-toxic compound. And both of these are quite high enough. And the average of these two is what gives you the balanced accuracy as well. The F1 score is a harmonic mean of precision and recall. So as a harmonic mean, you get both of these rates together in some sense, and you get an F1 score of 0.5, which is pretty decent. And on the second hand, after this, you try to calculate the ROC score, and this basically tries to put the true positive and the false positive rate together at different thresholds. So when I was talking about different thresholds, the threshold we chose here was 0.5, but there's possibility to choose the different threshold. And at different threshold, you have different points here, and you would get a curve. And the area of this curve is another metric. So area under curve of the ROC is 0.85, which is also quite good. 0.5 is generally a random model, which goes from this corner to this corner, and 0 would be a model that is worse than a random predictor here. So 1, which should be a complete vertical line and a horizontal line, would be the perfect predictor. Finally, I'm trying to understand in this part what the classifier model actually works on and how does this work. So we look at the visualization of a random forest model. So we have installed a particular module called dtree-viz, and this is a deep tree visualization, and we are trying to visualize this. So the function basically takes the first estimator. So out of random forest, all the trees, it selects the first tree, and with the first tree and the training data, we're trying to see where exactly the splits occur. So I'm going to try and open this in a new page. Does it work? No, but anyways, you can zoom in. So as you can see, what the model did here first, it shows the 1122nd fingerprint and said if it's 0, go this way, and if it's 1, go this way. And within those compounds which were 1, there were 19 of them which were having zeros again for this particular fingerprint. So for each fingerprint, it starts splitting the data, and it goes up to a depth of 5 because that's what our parameters said. Once we go to a depth of 5, for example, here, there's Morgan fingerprint 1522, and out of all the ones that were 0 here, a lot of them were actually toxic compounds. And over here, out of all the ones that were 1, a lot of them were sorry, now other way around. A lot of them were toxic here as well as toxic here as well. So for both cases, we predicted them to be toxic. But in this case, where you see it's coming here, for this particular Morgan fingerprint, if the bit vector 0 had a lot of compounds which were non-toxic, the model for any new compound which falls through this loop and comes here would predict the maximum possibility death. So any molecule that goes down this chain with these conditions and falls any of these places would be predicted as toxic. But if it falls on this one, it would be predicted as non-toxic. If it falls on the other one, it's predicted as toxic. So it keeps going on like this until a node is reached. You can choose to keep going this on until like the nodes become completely pure, and then the depth is basically more than 5. It reaches up to 20 or something. So that's one way to, but you stand at a chance of highly overfitting the model. So a depth of 5 is generally more interpretable in some sense. And next we look at feature importances. Next we look at feature importances, and feature importances gives you individual values for each feature from a random forest. And so if we have around 100 features which were the most important ones, so we have sorted those feature importances according to their value. So each importance comes from the classifier, and then we sort these by descending order. So the highest importance at the top, and I'm only showing the first 100 of them. So if we plot them together, you would be able to see which feature had the highest, say, importance for the machine learning model. In this case, it helped the mean decrease in impurity, basically. So it goes on like this, and these were less important and these were more important. However, do take into account that feature importances from random forest are subject to correlation effect. So if there were a lot of these features are very similar, they share their importance together. So taken together, if these 5 features are very similar, they have actually divided their importance among each other, which is why it looks like these 5 features are less important than this one, but it might be that there are a few features here which are very similar, and therefore their importance got diluted a bit. But overall, this is how a classification model usually works. You train a model, you look at the metrics. Mostly you look at the confusion matrix, the ROC, and the balanced accuracy are a few ways. F1 detection, if you are really interested in the active component or the minority class. Average precision score is another way as well. That would be all. And if you have any questions, please let me know. Thank you so much.
That's great. Thank you so much, Srijit. Yeah, thanks so much, Srijit. I think that's I realised that was a quick whistle-stop tour through modelling some mitochondrial toxicity data. I think it was really powerful in what you showed us there in terms of how we can take some simple initial classifiers and then we can ultimately end up with decision tree that ultimately can potentially guide us in the drug discovery process to avoid some of those compound liabilities if necessary. So that's really nice to see. So I don't know at this point if anyone in the audience has any particular questions to ask us, but I'd certainly like to say thanks to Srijit, to Layla, and to Andreas for joining this session today. I think it's been really important. I think we've learned a lot. I certainly have. It'd be great to hear some feedback from you guys. So perhaps you might want to send us an email, or indeed, I know that Layla, Srijit, and Andreas all have Twitter handles, for example, and they have GitHub repositories which are shared in that Google Colab. So yeah, do have a look at their repositories and see what other interesting models they're working on, and they've got their publications linked there as well. So yes, just to paste this back into the box. Where am I? To put this into the chat box. If you go to YouTube and type in this name here, which is a handle, this is most likely where the recordings will be deposited in the next couple of weeks. So yeah, please have a look. Layla and Srijit have posted up there some links to be able to contact them, directly. So please do. But in the short term, if there's no questions, then I suppose we can wrap up. Layla or Srijit, do you guys have any comments at all?
I guess thank you everyone. Thank you everyone for listening and for talking through this. So obviously we kind of gave you like the basic introduction. So hopefully we can have workshops in the future where we're going to go a bit deeper and go into a bit more detail. So yeah, we'd love to hear your feedback and yeah, hope to see you potentially in later workshops.
Great to see you as well. And yeah, feel free to contact us if you have any queries as well. Thank you.
Grand. Well, in which case then, I suppose we'll close down the session. But yeah, thanks everyone again. Thanks everyone for dialing in. We've had a really good retention rate. We've still got about 60 people on the call at the moment, which is really good. So I think that shows there's an appetite here. So I think as Layla said, maybe we might have some more of these sessions in the future. And hopefully have some of these kind of learning sessions again. So yeah, thanks again.